Good morning, everyone. We are now approaching the second day of the Metabolism and Cancer International Symposium, and we hope you have enjoyed yesterday. So today, as the first speaker, we have the pleasure to introduce you Professor Marcus Oliveira, who is an associate professor in the Institute of Medical Biochemistry at the Federal University of Rio de Janeiro. Marcus has an habilitation in biochemistry and molecular biology granted by the University of São Paulo and a postdoctoral research in endocrinology and metabolism at Boston University School of Medicine. His research program lies at the intersection of bioenergetics and cell biology, exploring how structural and functional modulation of mitochondria shapes the cellular responses to stress signals, including those generated by nutritional imbalances, immune activation, or aging. More recently, he has some studies with cancer as well. So we have to thank Professor Marcus for accepting to be here, and please feel free to go ahead when you feel ready.
Thank you, Gabriela, for the introduction, and also I want to thank Hobson and Luis for the kind invitation to present this project in the Metabolism and Cancer Symposium. So today I'll talk about the metabolic effects of mild iron overload in central and peripheral nervous system tumor cell lines. This project, in fact, was developed by Ana Paula Mendonça, my former master and PhD student. And over the years, my laboratory has been working on blood feeding organisms and trying to understand how blood feeding parasites and insect vectors of tropical diseases deal with blood. And one of the interesting things that we found out over the years is that the presence of blood derived products cause major changes on energy metabolism pathways. So keeping that in mind, we try to ask the question whether the presence of blood derived products such as iron, heme, and hemoglobin would also cause effects on metabolism in human cells. So this was essentially the question that Ana Paula tried to answer using the two tumor cell line models to answer these questions. So just to give an outline of this presentation, I'll firstly give a brief background on cancer metabolism and mitochondrial and iron metabolism in cancer. Then I will present some of the results we had on the consequences of iron metabolism on neuroblastoma cell line. A little bit further on the effects of iron on glioblastoma metabolism, and then some general conclusions to finish this presentation. So when we think on cancer, we always think on the changes that cells, normal cells, had to undergo to be transformed in a tumor cell changes. So there are some features that tumor cells have that make them unique. For example, the presence of genomic instabilities and mutations, the ability to induce inflammation triggered by their own tumor cells, the ability to evade the immune response to avoid destruction of the tumor cells, and the changes on the cellular energy metabolism. So particularly in this aspect, with looking at the literature, we see that the complexities that underlie the changes in the energy metabolism pathways are so complex, are so diverse and heterogeneous that makes very hard to understand exactly, to pinpoint specific mechanisms for specific tumor cell lines that make them unique. And particularly on this aspect, it's impossible to talk about changes in energy metabolism without considering where these observations came from. And this came firstly by Otto Warburg, that in his seminal studies, he found out that the cancer cells were able to maintain ATP production mostly by anaerobic mechanisms instead of the aerobic mechanism. So the tumor cell lines are basically maintaining the energy provision by anaerobic pathways, which is distinct from normal cell lines, which is coined the term of aerobic glycolysis or the Warburg effect. But over the years, this concept that the tumor cells are essentially Warburg dependent changed a lot, especially in the last years when by using omics approaches, that eventually found out that the metabolism of tumor cells are heterogeneous and flexible, depending on the changes in the environment and in the nutrient supply. So today, when we think on cancer metabolism, we have to consider the heterogeneity nature of the tumor cells. Not all tumor cells are identical, and they are different. They have different energy metabolism pathways that are required to maintain cell viability. But they are also flexible. Even the same tumor cell line can change its metabolism depending on the signals that the cells receive from the environment. For example, depending on the cell cycle, the differentiation state, eventually xenobiotics that generate changes in the genome of the cells can cause changes in energy metabolism, but also the nutrient availability and interactions, for example, with extracellular matrix. So all of these underlies what we have to consider when one has to work and think on the changes on the energy metabolism during cancer cells. Specifically, on the topic of this talk is the neuroblastoma. And the neuroblastoma is a particular kind of cancer that is very frequent in children, and it's responsible for close to 15% of childhood cancer deaths worldwide. And the neuroblastoma is a tumor that is derived from the sympathoadrenal precursor cells that, upon the activation by DNA methylation, histone modification, and other factors, these cells can migrate to adrenal glands and then differentiate to a tumor cell in the adrenal glands. So these tumors can be very severe, and they are very important in terms of epidemiological perspective. So these cells are those that will generate the tumor, the neuroblastoma. And the neuroblastoma has a unique metabolic profile. A recent study has identified in 23 neuroblastoma patients, pediatric patients, that were infused with radio-labeled glucose and measured the metabolic profile, the metabolome from the tumors, and also performed tracing studies in these patients. And what this study found out is that the neuroblastoma has a unique metabolic profile, completely different from many other cancers, kind of cancers. Basically, one of the features that they found out is that the neuroblastoma has a higher lactate production derived from glucose compared to other tumors. And not only that, but also the production of dopa metabolites is also a hallmark of neuroblastoma. Beyond that, we had to think on iron metabolism. But why do we have to think on iron metabolism? Iron is a very important element for any cell for different reasons. And usually in mammalian cells, iron is transferred to cells through a transferrin mediated mechanism that upon binding to the transferrin receptor, the iron is transported through endosomes and then released in the cytosol, comprising what is called the labeled iron pool. This labeled iron pool can be used for different cellular purposes. For example, DNA synthesis and repair, production of iron sulfur clusters and heme metabolism, and also stored as ferritin. In conditions where iron levels increase, iron can be exported to a ferroportin hepcidin mediated mechanism, maintaining the labeled iron pool stable. However, in cancer cells, the scenario is very different. And over the years, the literature have pointed out that changes and increases in the iron levels are directly associated with cancer occurrence and severity and risk of cancer. And these evidence are supported by different observations that support that the labeled iron pool in cancer cell increases by two basic different mechanisms. One, by the increase of transferrin mediated receptor expression, and the second, by a lipocalin mediated iron transport that increases the labeled iron pool together with a reduction in ferroportin uh, exporting iron mechanism. So all of these changes results in increase in the labeled iron pool. And in the neuroblastoma specifically, uh, although the iron deficiency is observed earlier on diagnosis, over the course of the disease, several markers of increased iron levels are observed. For example, increased in serum ferritin associated with disease stage and severity. And at the same time, iron chelation therapies has been proposed as a mechanism to limit neuroblastoma growth. And thinking on the context of the cellular iron deposits, we know that the mitochondria are the major cellular sites of iron storage. For example, in intestinal epithelial cells, mitochondria is the dominant iron storage site, while in the lymphocytes, almost 55% of cellular iron is found in the mitochondria. And once in the mitochondria, how is the iron handling in the mitochondria? So iron is taken up by mitoferrin, which is an iron transporter located in the inner mitochondrial membrane. Iron is transported to the mitochondrial matrix, and once in the matrix, iron can be used by ferrochelatase to produce heme, or iron sulfur clusters by other mechanisms that support, for example, electron transport chain activity and other iron sulfur clusters in the cell. Or iron can be also stored by a specific mitochondrial ferritin. But these effects, how are the effects when changes in iron levels happens in the cells? So the changes depends on the way that iron is administered to mitochondria or to the cells or to the organism. For example, if iron is topically added to the mitochondria, different studies have observed increased or reduced respiration rates in different tissues, reduced mitochondrial membrane potential and oxidative stress. But changing not for topic, but for acute or chronic iron overload, it seems quite clear that there's reduction in ATP production by mitochondria, and also increased proton leak and oxidative stress. And beyond that, iron also changes the mitochondrial reticuli. Morphology of mitochondria significantly changed. For example, in primary neurons, mitochondria is clearly fragmented when exposed to iron. But what are the metabolic effects of iron in neuroblastomas? There's a little information available on this topic. But this study, interesting study, published a few years ago, have demonstrated that 500 micromolar of iron hardly changed cell viability, the neuroblastoma viability, indicating that neuroblastoma cells are surprisingly resilient to iron overload. And more surprisingly, the levels of mitochondrial oxidant is not hardly affected, indicating that despite the fact that iron is a pro-oxidant by nature chemically, this is the cells, the neuroblastomas, are not affected in terms of the redox balance, at least in mild exposure to iron levels. So the working hypothesis that we raised was that the exposure to mild iron overload would change mitochondrial and redox metabolism in a way that this would associate with neuroblastoma growth. So our main aim was to investigate these effects using SY5Y cells from neuroblastoma. So what Ana Paula did, Ana Paula used this this neuroblastoma cell line in these conditions, and specifically doing iron sulfate heptahydrate treatment for 24 hours, and then measure different readouts, cellular, molecular, and biochemical readouts to address these questions. So the first thing that we observed was to assess the viability of these cells upon iron treatment. So we can see here that the morphology, the general morphology of the neuroblastoma is not affected. The cytotoxicity is maintained. The protein content of the cells is preserved. And the viability assessed by MTT reduction is also not affected at 100 micromolar. So we used this concentration of iron overload in all the subsequent experiments. And just to make sure that the treatment resulted in increased iron levels in the cells, we measured the total iron levels in the cells by IBqM mass spectrometry. And we see an increase in almost fourfold increase in iron levels in the cells. So these observations indicate that the neuroblastoma are in a way resilient to iron exposure, at least for these concentrations. So once establishing these conditions and making sure that the cells are not dying, we then move on to assess the effects at the cellular level. So the first thing that we analyzed was the proliferation and migration ability of the neuroblastoma upon iron treatment. So we can see here that the migration, the capacity of the cells to migrate was significantly reduced upon iron treatment. And surprisingly, the cell proliferation strikingly increased, suggesting that at this condition, the cells shift towards a more proliferative instead of migration, which is in a way similar to many other changes caused by other drugs and other stimuli in cancer cells. So in a way to address whether the iron would affect energy metabolism and specifically mitochondrial metabolism, we then measured whether the iron treatment would accumulate within neuroblastoma mitochondria. And to do that, we used an approach using the fluorescent probe mitochondria, which is a cell-permeable probe that is uptaken by mitochondria. And once in the mitochondria, the iron levels modulate the fluorescence ratio of this probe. So higher the fluorescence, higher the iron pool in the mitochondria. And we observe here that in these conditions, the mitochondrial iron levels increase substantially, suggesting that the iron that is taken up by the neuroblastoma is eventually reaching the mitochondria. So then our next question was, what are the consequences for the morphology, content, and function of mitochondria in the neuroblastoma upon iron treatment? And to do that, we measured the fluorescence intensities of mitochondrial green as a readout of mitochondrial content and mitochondrial form, and also mitochondrial membrane potential by TMRE fluorescence. And we can see here, so this is the control cell, these are the control cells, these are the iron treated cells, and these are the control cells exposed to a proton ionophore, FCCP, that is known to collapse the mitochondrial membrane potential. So we can see here that the intensity of mitochondrial green is even increased in the iron treated cells, and the intensity of TMRE as a readout of mitochondrial membrane potential is strongly reduced in iron treated cells. So we quantified these effects. So the content of mitochondria measured by two different approaches, one by the mitochondrial green integrated intensity, we see there is no significant changes in the mitochondrial content in the iron treated cells. And this was corroborated by quantitative proteomics that I'll talk a little bit further in this presentation. When we quantified different mitochondrial proteins, such as prohibitine, succinate dehydrogenase, subunit A, and cytochrome oxidase subunit 2, we don't see differences, suggesting that the mitochondrial content is not affected. But the form is affected. And we can see here by measuring the circularity of the mitochondrial green signal. So the circularity is a measure of mitochondrial, is a measure of form in a way that when an object is closer to one means a perfect circle. So in this observation, we can see that the iron-treated cells, the circularity is significantly higher compared to controls, indicating that the mitochondrial fragmentation is induced by iron treatment, as is known for non-tumor cell lines. And the quantification of membrane potential, we can see that there is a significant reduction of mitochondrial membrane potential. So these changes in membrane potential are caused by increased mitochondrial iron levels without affecting the total mitochondrial content. In a way to have a further detail about a mitochondrial function and changes induced by iron, we then move it to assess oxidative phosphorylation capacity of the cells by high resolution respirometry. And just to brief background on this powerful technique that is able to assess the mitochondrial function even on intact cells, we can measure the oxygen consumption rates of the cells over time. And by using pharmacology with different drugs, we can assess different metabolic states in the mitochondria. So the basal respiration or the respiration of the cells are doing in normal conditions. We can assess the ATP-linked respiration, which is part of the basal respiration that is inhibited by oligomycin. The proton leak respiration, which is the remaining respiratory rates of the basal respiration that is not inhibited by antimycin and rotenone. the maximal respiratory capacity, which is green box, which is the highest respiratory rate that any cell can perform when there is no limiting effect of the protomotive force, and the spare respiratory capacity. So the spare respiratory capacity is the part of the respiratory rate that is the difference of the maximum minus the basal respiration, or in other words, how much the cells can increase their respiratory capacity when the energy demand increases. And these are powerful measures that we can assess the function of mitochondria even in intact cells. So for example, we can observe, for example, changes in the basal respiration, for example, an increase. If we observe an increased basal respiration, this could be a consequence of either an increased ATP-linked respiration or oxidative phosphorylation capacity, and or an increase in the proton leak. The consequence of this is that the ability of the cells to respond to an increase in an energy demand measured by the spare respiratory capacity will be reduced. So with this in mind, we then assessed the mitochondrial metabolic effects of iron-treated cells on intact cells. And then we can observe it here that the basal respiration slightly but not significantly increase. The same for OXPHOS or the ATP-linked respiratory rates, the proton leak respiratory rates. The maximum respiratory rates are not changed at all, but the spare respiratory rates are significantly reduced. And this is very interesting because this indicates that somehow the increased mitochondrial iron in neuroblastoma is reducing the capacity of neuroblastoma to increase their energy demand if the energy demand is somehow increases by different stimuli. If we normalize this respiratory rate by the mitochondrial content, by dividing these values by the mitochondrial green intensity, we see a similar pattern. So no differences in all metabolic states, but the spare respiratory rates are significantly reduced. So these observations indicate that the changes that the mitochondria of neuroblastoma cells undergo increases slightly the basal respiratory rates, both by a component of increased ATP-linked respiration and increased leak. Is not significant, but it's robust. That ultimately impact the spare respiratory rate, or the capacity of the cells to increase respiration if energy demand increases. Another way to look at this data is to normalize the metabolic states by the maximum respiratory capacity of the cells. That's the ratio of the basal, leak, and OXPHOS ratio over E, which means the maximal respiratory rates of the cells. And we can see here that there is no difference at all in all metabolic states when we consider the maximum capacity of the cell, suggesting that even with those mild changes on the metabolic profile in the mitochondria upon iron treatment, this is not affecting significantly the capacity of the neuroblastoma to produce ATP by oxidative phosphorylation. But what are the other components that might also be affected by iron treatment of the cell? And then we look at the other components in the redox perspective. So we know that iron is a free radical by chemical itself. Iron is a pro-oxidant. There are many evidence that support that. And we decided to measure the total oxidant levels of the neuroblastoma by using the dehydroetidine fluorescence as a readout of oxidant levels. And we can see here the control cells, the beautiful signal of DHE, indicating that these cells are producing oxidants. But surprisingly, upon 24 hours of iron treatment, we see a strong reduction in the total oxidant levels, which is quantified here. And this was quite surprisingly for us, because we would expect that iron, usually almost all the literature at the cellular context puts iron as a pro-oxidant, and it is a pro-oxidant. But in most of these studies were carried out in the presence of huge iron concentrations. And at this case, it is different, because in this case, we are just mildly changing the iron levels. And the response of the cell upon iron exposure, in a way, mimics not a preventive antioxidant mechanism, in a way to reduce the possibility of iron to interact with reaction oxygen species and then boosting oxidative stress. So we decided to measure oxidant levels using a different approach by measuring the total hydrogen peroxide rate extracellularly. So we measure the total hydrogen peroxide produced by the cells. And we can see here that this, again, there was a strong reduction on the hydrogen peroxide productions of the neuroblastoma upon iron treatment. So this indicates that this mild iron overload triggers an antioxidant preventive mechanism to avoid oxidative stress, which is very interesting for us. And we decided to further explore these changes in the neuroblastoma by performing quantitative proteomic analysis of these cells. So we quantified the total proteome of the neuroblastoma treated cells, and more than 4568 proteins were quantified, and establishing a 10% cutoff of fold change, increase and reduction of these proteins. We eventually reached 2,404 differently expressed proteins that most of them were downregulated. I will not talk about them today, but I'll concentrate on this on the top 662 proteins that were upregulated by iron treatment. Some of these candidates were expected. For example, we know that any cell that is exposed to iron overload has a specific phenotype in terms of gene expression. For example, transferrin receptors are downregulated at the same time that ferritin expression increases, in a way to reduce the possibility to increase the labeled iron pool in this iron overload condition. This is exactly what we observed, as the ferritin light and heavy chain increases and the transferrin receptor reduces. So this is a very good control for us to indicate that the cells, the neuroblastoma, are responding as expected in terms of the iron metabolism. One of the most striking candidates that pop up from the quantitative proteome was this protein, prominine 1. Prominine 1 was upregulated by more than 90 times upon iron treatment. What is prominine 1? Prominine 1 is a plasma membrane glycoprotein that is associated to many cancer types of cancers. And in the neuroblastoma specifically, previous studies have found out that the silencing of prominine 1 reduces colony formation and neuroblastoma growth in an animal model, suggesting that the prominine 1 somehow is associated to neuroblastoma growth, which is it's strongly indicating that this increase in PROMINI1 might be an underlying mechanism for the increased proliferation that we observed upon iron treatment. Another interesting protein that popped up was prolyl hydroxylase, which is increased 23 times upon iron treatment. Prolyl hydroxylase, as many of you are aware, is a protein that is activated by iron and promotes the hydroxylation of hypoxia-inducible factor that is eventually degraded by ubiquitin system. So this indicates that the increase in the prolyl hydroxylase expression might increase HIF-1 alpha degradation. And once HIF is degraded, mitochondrial function is preserved. So this is very interesting to link as a potential target for further studies in our laboratory. Another interesting target was sideroflexin 4, which is almost 20 times increased. Sideroflexin 4 is directly involved in iron sulfur cluster and heme biosynthesis, which it might be linked to our phenotype. We also observe an increase in TLK1, a serine protein kinase that is involved in DNA repair and has anti-apoptotic roles. Twinkle is a protein, mitochondrial protein that is directly involved not only in mitochondrial DNA replication, but also in restoring mitochondrial DNA damages. So this is a very interesting target. We also observed the slight increases on mechanisms involved on mitophagy, on a specific type of mitophagy, such as BNP3 and NIX, which targets mitochondria to undergo autophagosome degradation. And finally, two different mitochondrial proteases, CLIP and LON. And CLIP and LON are part of the mechanisms for mitochondrial proteostasis, to degrade damaged or misfolded proteins as a mechanism for mitochondrial quality control. And looking further on these two proteases, we were initially interested in these two proteases. Looking back in the literature, we saw that in different models, that when CLIP is silenced, the viability of the cells is reduced, and reduction of viability in CLIP silencing is associated with both reduction in respiratory rates and increased ROS, mitochondrial ROS production. But what are the targets of CLIP? One of the important targets of CLIP is this subunit of mitochondrial complex 1 and the NDUFV2. This protein is one of the first targets of CLIP. And this complex 1 subunit is one of the most important sites of superoxide generation in the mitochondria. So we look back on the quantitative proteome to check whether NDFU2 would be affected by iron treatment. And we can see here that there is a complex scenario here. Some proteins are downregulated by iron, and some are upregulated. But we can see that NDUFV2 is one of the proteins that are significantly reduced upon iron treatment. So this suggests that increased CLIP and LON expression might be involved in the NDUFV2 degradation that would explain the reduced superoxide production in iron-treated cells. But beyond these changes, we also observed that there was a significant increase on the 6-and-8 dehydrogenase subunit D expression. And then we asked the question whether the iron treatment would change somehow the electron transport system complexes in a way to shift the electron donor capacity from complex 1 to complex 2. And then we initially tested this possibility by challenging the iron-treated cells with a classical complex 2 inhibitor, malonate. And we can see here that the viability of neuroblastoma cells treated with iron is significantly reduced when they are co-incubated with malonate, suggesting that the phenotype, the viability of the neuroblastoma in the presence of iron somehow involves an increase in the dependency, an increased dependency of 6 and 8 dehydrogenase expression or activity. So at this point, we can establish some connections, and we can see that the changes that we observed upon iron treatment involved the reduction of an increase in prolyl hydroxylase expression that might involve a reduction in HIF1 alpha to maintain mitochondrial structure and function. We also saw an increase in NIX, BNIP, twinkle, and CLIP-LAM protease systems that are directly involved in selective mitophagy, mitochondrial DNA stability, and mitochondrial proteostasis. In the case of selective mitophagy, we think that this might be a mechanism to contribute to reduced ROS levels. And the mitochondrial proteostasis might be associated to reduced NDUFV2 expression and complex 1. That might also contribute to reduced ROS production. At the same time, that we have some evidence that see a shift towards a more complex 2 dependent mechanism to maintain ox-force and energy homeostasis. But also, we still don't have a direct explanation for these effects, but we think that this complex 2 dependent mechanism might be also contributing to reduced ROS, and ultimately reducing also a slight reduction in mitochondrial membrane potential that will ultimately reduce ROS production. And all these mechanisms can be associated by still elusive ways by involving prominent 1 and TLK1 proteins that eventually associated to a higher proliferative and lower migration capacity of the neuroblastoma. So moving quickly to the second part of this presentation, I'll talk briefly in the next few minutes to the metabolic effects of iron on the glioblastoma context. And we know that for the glioblastoma, the iron metabolism is very important, as clinically glioblastoma are highly associated with intracranial hemorrhage, in a way that the glioblastoma cells are much more exposed to blood-derived products, such as hemoglobin, heme, and iron than in the neuroblastoma context. So Ana Paula asked the question, whether iron would also affect energy metabolism in the glioblastoma cell line. We used this cell line, U138 cell line, using the same approach that we used for a neuroblastoma, and then measuring different readouts. So again, we look at the morphology of the cells treated with 100 micromolar of iron for 24 hours, and we don't see no observable difference in the morphology. We saw twice increased levels of iron in the intracellular iron in the glioblastoma. No significant differences in the viability and the protein content of the cell. So in these conditions, we see that the iron is not causing cell death. But remarkably, when we look at the mitochondrial structure and function, we can observe here by the mitotracker green and TMRE fluorescence that the mitochondrial content is not that affected. So the mitotracker green signal is not significantly changed, but we can clearly see here this remarkable changes in the mitochondrial morphology. And we can measure here by measuring the circularity of the mitochondrial signal. We can see that there's a significant increase on the circularity of the mitochondria. And we also observe the reduction of mitochondrial membrane potential. So these metabolic phenotype is quite similar to those that we observed in the neuroblastoma. So looking again on the mitochondrial function by high resolution respirometry, we can observe here that there's no significant changes in all metabolic states of the glioblastoma cell line, but in this case, a significant reduction and the maximal respiratory capacity of the cells at the uncouplet state. So different from the neuroblastoma, the glioblastoma has a direct effect of the maximal capacity of the mitochondria to reach the maximal respiratory rates. That is a slightly different scenario from the neuroblastoma. And when we look at the redox side, this was again striking for us that the cells that undergo iron treatment, we can see here by the DHE fluorescence that the total oxidant levels are reduced by iron exposure, which is quantified here. And when we measured the total oxidant production by an independent measure of extracellular hydrogen peroxide levels, we can see that this exactly the same, suggesting that the responses that the neuroblastoma and glioblastoma undergo upon iron treatment converge to a reduction in the cellular capacity to produce oxidants. That involves a reduction in the membrane potential, a preserved mitochondrial content, and slightly changes in their functions. So to conclude, the take-home message that I want to leave you here is that the mild iron overload in these two different tumors, the neuro and the glioblastoma, promotes a metabolic rewiring that preserves oxidative phosphorylation capacity of the cells and reduces oxidant productions. And we think that the combination of these two consequences are critical for the differences in cell biology that we observed, including increased proliferation and reduced migration of these cells. So I finally want to thank the people involved in this work. First of all, of course, Ana Paula, that made almost all the data that are shown here, but also the people in my lab that were directly and indirectly involved in these studies. I would also thank the collaborators that were critical, and especially a special thanks to Rodrigo Rossignol and Nívia Amoedo at the University of Bordeaux that performed many experiments that I shown in this presentation, and the funding for this for my laboratory. And I just wanted to leave a message that I think is very important for me personally, is that this work would not be possible without Franklin Hunjanek, which is he was one of the most gifted minds I ever have the pleasure to share my career with. And I want to dedicate this presentation for him. Thank you.
Thank you very much, Marcus. It's really good to see Franklin in your presentation. It's really good to see that you're working with cancer, so fantastic. So, yeah, Great talk. We have the first question here from Gustavo. Let me just check. Okay. Amazing talk, Marcus. Prominent ACA CD133 is related to cancer stem-like cells in glioblastomas. Do you think that this iron mechanism is associated with differentiation?
Yes, I think it's possible. We are currently conducting experiments to silence prominent 1 in the neuroblastoma and check. One of the readouts that we want to look at is exactly the shift of proliferation and migration. And we think that the result that we're going to have is a reduction in the proliferation, but I'm not quite sure in the migration of the cells. I also want to stress that in all these studies, in the neuroblastoma, we didn't differentiate the cells with retinoic acid. It's important to notice that, to stress that, because this cell line specifically is usually people in different labs differentiate the cells with retinoic acid. All the studies that we've done here were in undifferentiated cells. So it might be the case, yes.
Okay, so we have a question from Alicia. Very nice talk. I guess a lower steady state oxidation of DHE and hydrogen peroxide released from cells is expected if ATP linked and leak respiration is higher, although we don't see this in glioblastoma.
Yeah, great question, Alicia. Yes.
Yeah. And the question, I think it comes next, but if you want to make a comment, it's fine. Okay. And her question is it possible that there isn't a fast oxidative burst before measurements leading to the damage that promotes these respiratory changes?
Yes, great question, Alicia. We think that it might be the case because iron is a pro-oxidant. Iron is a free radical by itself. So, and there's an increase in the mitochondrial iron upon the treatment. So it's possible that a slightly redox burst early on treatment might trigger the phenotypes that we observed. One of the experiments that we want to address is to treat the cells with mitochondrial target antioxidants before iron treatment to check whether these early bursts might be causing the observed effects. And the first question is that if these early oxidant bursts take place, I would expect an increase in proton leak respiration, as these would reduce oxidant production by mitochondria by slightly reducing membrane potential. Well, I'm not hearing.
I'm sorry.
Oh.
Also, her last question. Also, did you measure glutathione in reactive nitrogen species as added iron could affect dinitrosyl iron complex formation? This could explain the protective redox effects of iron.
Yes, we have experiments. I didn't show here, but we need to repeat this experiment. That's why I didn't show it. But total glutathione and glutathione balance reducing an oxidized glutathione doesn't change upon iron treatment. And also the expression levels of the main antioxidant proteins, enzymes, are not affected at all by iron treatment. So we don't think that the phenotypes we observed are a consequence from the redox perspective, are not consequence of repair of damaged oxidized damaged molecules or by antioxidant systems, but rather by preventive antioxidant systems. So by mild proton leak, I think this is a potential explanation for these effects.
Okay, we have many congratulations in the chat. The next question is from Juan Perez. Hi, Marcus. Nice presentation. Did you incubate the cells with succinate to test your hypothesis? Prostate cancer cells have the ability to oxidize succinate, but this is not related to proliferation.
Yes, we didn't test the succinate yet, but we measured cytochrome C reduction provided by succinate, and we don't see a, a slight increase, but we think that there are other phenotypes in the mitochondria that might explain this. But I wouldn't go further on this because we don't have much support. But I agree that this is a nice venue to explore the role of succinate in this scenario.
Great. And now we have a question from Marie-Pierre. Thank you for this very interesting work. Do you know for how long are iron overload effects maintained? Are they reversible?
Yes, a great question. No, we don't know for how long the neuroblastoma and glioblastoma retain iron, but we think that these cells can retain iron for a longer time, because when we incubate the cells for longer times, 48 hours and 96 hours, some of the phenotypes are still maintained. So we think that the cells eventually maintain the iron for longer times.
Okay. Now I have two questions, particularly concerning the first part of your talk. So I don't work with iron metabolism. So at the beginning, I was wondering what was the concentration of iron, intracellular iron, that would be expected for those experiments that would reach the cells in a normal or in a cancer condition. Like the concentrations that you have used in the beginning are do resemble this environment in a normal or in a cancer setting?
Yeah, it's hard to define exactly because the cellular iron levels in the neuroblastoma in a patient varies over time. So it depends on the grading of of the tumor stage and eventually the treatment. So we decided to choose a concentration of iron. Our main concern was based on the fact that iron is a pro-oxidant and iron can't kill the cells.
Yeah.
So we decided to choose a concentration that was at the limit at the edge of cell death. So that was critical for us to avoid any of the effects that were resulted from apoptosis or necroptosis or ferroptosis, that I even talked about that, because we try to avoid that to make sure that this exposure is not damaging the cells, but rather iron would act as a signal rather than a toxicant for the cells. And I think most of the literature sees iron as a devil that kills the cells, but that is not the case. Mild changes in the iron availability might also have clear effects on the cellular and the metabolic responses from the cells.
That's
So
great. That was exactly my point, because I was wondering, like, which concentration would shift to the ferroptotic state, because we always think about the therapeutic perspective, and you're thinking about the metabolic perspective, and that's great. Yeah, you got the concentration that could not induce cell death, and instead could like induce metabolic changes. So interesting, very interesting.
Yeah, and just to complement my answer is 100 micromolar of iron for a normal cell can kill most of the cells. So these tumor cells that I work with are remarkably resilient to iron. So this underlies the higher ability of tumor cells to handle iron. It's a hallmark of one of the hallmarks of cancer.
Yeah. And another question, do you pretend or think about like performing those experiments in a 3D setting, like 3D structures that would better resemble the oxygen gradients and the spatial architecture of solid tumors?
We didn't consider this yet, but yeah, it might be very interesting to consider this possibility in the context of hypoxia, you mean, right?
Yeah, and taking into consideration the heterogeneity of neuroblastoma, maybe you could like get different metabolic states within the tumor, like depending on if whether this iron, iron concentration at the core or at the border of the tumor.
Yeah, this is a very interesting follow-up. But yeah, we don't have the expertise. My laboratory doesn't have the expertise for that, so I'll definitely have the need help from my collaborators to do that, because we are a mitochondrial-based lab. So I'm a newcomer in the field of cancer. So
Yeah, we've never
pardon
performed with
me for my.
neuroblastoma cells, but like with breast cancer, we have some kind of expertise,
Yes,
early
let's talk
expertise.
about
So
that.
maybe we
I'll
can
be
talk
very
more.
interested too.
Yeah.
Yes, thank you.
Okay. Let me check whether we have further questions. No, it's just Hudson and Amanda. Like, congratulations on your work and your presentation and the talk. So
Thank you.
I think we don't have further questions. So I should thank you again for being here and sharing this data, very exciting data, new data. So
Thank you, Gabriela, for the
yeah,
opportunity.
that's great. We hope we can see you in the near future in other cancer events. That would be great.
Yeah, thank you.
Okay. So for the audience, like we're gonna have a just quick break so that you can stretch your legs, but be tuned. We'll be back like in 5-10 minutes. So stay tuned. Okay. Bye bye.
Hello, everybody. I hope you can hear me. Thanks for coming back for our second day of our symposium. And I'm here to introduce you the next speaker, which is an honor to introduce her and a pleasure also. And I'm very happy to have her here today because we have worked very closely in the past. And I hope we still continue to work together, which is Marie-Pierre Junier from the Inserm in Paris, France. Well, Marie-Pierre has a very beautiful pathway on her career, and she started her PhD studying the discovery and the characterization of lipid mediators that regulate the activity of hypothalamic neurons. And for her postdoc, she worked with Dr. Sergio Ojeda in Portland at the USA. And in this postdoc, they provided the first demonstration that growth factors play a very important role in the control exerted by the brain on the initiation of puberty in both normal and pathological conditions. Well, when she finished her postdoc, she went back to France and she was recruited by the Inserm, as I told you before, and she led studies on the role of the growth factors in neuroglial interactions, neurodegenerative diseases, focusing especially in the signaling mediated by the EGF receptor and its ligands. Well, and to further explore the role of EGFR signaling in glial cells behavior, she then joined the glioplasticity team at that time at the Jacques Glowinski lab at the Collège de France in Paris. And she then started working on cancer's transformation, which led her to focus on brain neoplasms. And in 2008, she took in charge the co-direction with Hervé Schneeweiss, the glioplasticity team and neuro-oncology. And now she's going back to metabolism, the first subject that she addressed as a young scientist, while entering the adventure of the big data studies. So Marie-Pierre, I'm very glad to have you here today. And so you're welcome to share your presentation. And hello, good morning. Good afternoon in Paris, right?
Yes, it's good afternoon. Yeah. So I'm just sharing my screen right away, and I hope everything is going to go well. And in the meantime, I thank you very much for your very nice presentation. Does it work? I hope so. Could you tell me?
It's
okay?
Good. Yeah.
Okay. Okay, thanks a lot. So thanks to all the organizers for giving me this opportunity to share with this very interesting symposium the last results we obtained in our team. And I'm going to talk to you today of how we try to handle and harvest big data in order to understand a little bit further the role played by metabolism in the control of the property of human glioblastoma cells. So I will go briefly about glioblastoma. I know that a lot of you are already aware of what they are, and you had a presentation by Hervé Schneeweiss yesterday. But glioblastoma are primary brain tumor that mainly affect adults. They are very aggressive. The half-life survival expectancy of the patient does not exceed 20 months, and that despite very aggressive treatment, which includes surgical resection whenever possible, associated with radiotherapy and chemotherapy. So these tumors are very heterogeneous. First of all, at the level of their genomic alteration, because from one patient to another, when you look at the whole tissue, you will detect varying predominant mutations. And of note, we do not know for sure the founding mutations of this tumor, because when the patient presents at the hospital, they are already with very huge tumor, and we have only some ideas and hypotheses about the cells of origin. We are sure that they come from the neural cells of the brain, and of the combination of mutation that could have resulted in the transformation of the cells. But anyhow, when the patient present, the tumor have mutation loads that can vary from one patient to another. They can also have present high or low copy number variations in their chromosome. And in the more recent years, people profiling the transcriptome, the whole repertoire of transcript in tumor tissues, have disclosed varying profiles that they designated as proneural, mesenchymal, or classical. So classical, for example, illustrating a predominance of an overexpression of the EGF receptor. When you look at each tumor, you can find also an extensive heterogeneity. This is first seen at the histological level because within each glioblastoma tumor, you have area that intermingles with different aspects. For example, some are very dense in tumor cells with a lot of angiogenesis. Others are on the opposite, deprived of the tumor, almost deprived of tumor cells and present big necrosis. And you also can encounter areas where tumor cells are more dispersed and are going throughout the otherwise normal brain parenchyma. But when you look further at the cellular level, you find out that the tumor is constituted by a mixture of normal and cancer cells. Next to the cancer cells, you have the normal neural cell of the brain, such as oligodendrocytes, neuron, or astrocyte, but also a lot of cells coming from the immune system, like the neutrophils, monocytes, and macrophages, and so on. And obviously, all the cells that make the blood vessels. Now, if we go even deeper and we look at the cancer cells themselves, we find out that those cancer cells are not identical, and that the tumor is made of different subpopulations of cancer cells. Data have accumulated in order to try to understand the sources of this heterogeneity in the cancer cell subpopulations. And the main source is, of course, linked to the dynamic of mutations that accumulate over the course of the development of the tumor, resulting in cellular clone exhibiting different genomic alterations that all coexist within the same tumor. But more recently, evidence have accumulated showing that another source of this heterogeneity is the capability of the cancer cells to adapt their functioning state to change in their microenvironment, whether these changes are related to the natural growth of the tumor, or even if these changes are related to the therapy applied to the patients. So as a result, within the tumor, you can have cancer cells in different functioning states, although they can present a cell with the same genomic background can present different functioning states, like, for example, a pro-angiogenic state, a proliferative state, or a migratory state. And all that results from this exceptional plasticity of the cancer cells that can go back and forth between different behaviors in order to adapt and survive all the evolution that takes place within the tumor. So here I listed some of these functioning states that a tumor can adapt, like, for example, tumorigenic or non-tumorigenic, that is able to initiate a novel tumor by itself, stem-like or not, proliferative or crescent, migratory or static, and we can also find cells which are either sensitive or resistant to therapy, and even cells that have adopted a senescent mode of functioning. And there are current works going on to better understand how these senescent cells participate to the tumor growth. So in our team, we decided to simplify this overwhelming complexity and to consider the functional state of the tumor cells in link with their underlying metabolic pathways. And we thought that through this consideration, we could find a way to overcome glioblastoma heterogeneity in order to pinpoint novel molecules that could offer therapeutic targets of great interest that is able to be efficient across a variety of genomic background. So why that? Just simply because metabolism, this array of enzymatic activity which is localized downstream of all signaling pathways that control cell functioning. So if we find something leading with key molecules of the metabolism that can affect this adaptation of the cancer cells, then we can hope to develop therapy that would be efficient for differing patients, but also for the same patient, for the different subpopulation of tumors, of cancer cells that populates his tumor. So, but to do that, we need to look at the system at the single cell level. So why? Because as I said earlier, if you look at the protein or transcript in a tissue, the results are an average of all the different transcript of protein levels that you observed in the tissue or even in the cell culture. And it could be also an average of the different mode of functioning of the cells or cells that have genetically differing background. And in the tissue, you even average the data over normal and cancer cells. So we decided to explore whether we could have obtained a systemic view of the metabolic pathway which are at play within the patient tumor in links with their cell functioning state using a data set of unicellular transcriptome that have been derived from the brain tumor patient directly after the surgical resection of their tumor. And of note, this series of transcriptome offer potentially the closest view that we can have to the tumor at the time of diagnosis, that is when we can access to the patient and offer him some therapies. So the analytical strategy with design was at first simple. We thought that we could analyze this differing transcriptome in order to extract information about the functioning state of the cells, then pinpoint the associated metabolic pathway prior to test the functional relevance of our finding using in vitro and in vivo models of human glioblastoma cells. Such a strategy relies on two postulates. The first one is that cell functioning state could be identified from single-cell transcriptome. And that's not that obvious because we all know that there is a world between the mRNA of a gene and its protein product, which is the final active product within the cells. And the second postulate was even bolder, that we could extract from this transcriptome profile not only the metabolic activity, but also their key enzymatic players. Up to now, the studies that have been performed on the single-cell RNA-seq data extracted from glioma cells from the patient have been focused essentially on different things linked more to the cell of origin of the tumor and also to its crosstalk with the tumor environment. The first data set that was ever published on glioblastoma cells from patients was published in 2014 by the Bernstein Laboratory at MIT. And this first analysis revealed that within a single tumor, we could encounter cells exhibiting all the transcriptional profile proneural, classical, mesenchymal, that had been first thought to distinguish subtype of glioblastoma. And here we see that it was, we found out with this publication that that was not the case. A bunch of other publications accumulated, all studying more the cell lineage and the developmental hierarchies that preside over glioma and glioblastoma development. And finally, and more recently, several other studies have focused on identifying the cell profile linked to the tumor and microenvironment and trying to reconstruct the crosstalk that takes place, for example, between glioblastoma cells and the immune cells populating the tumor. In our case, when we started, there was a single data set available, the one published by Darmanis and colleague at Stanford, and it comprised a little bit more than 1,000 glioblastoma cells, together with 2,500 normal cells, all coming from 4 different patient glioblastoma. So the first things that we did is to verify and determine whether a simple unsupervised analysis applied on these 1,000 transcriptome could help to group cells according to their cell functioning states. And this unsupervised analysis is very simple. It's based on principal component and analysis, and it's based on the overall similarity of the transcriptome of each cancer cells to its neighbors, with its neighbors. And what we found out it was that this unsupervised ion clustering analysis highlighted first the tumor from which the GBM cells were derived. You can see here on this principal component illustration that each dot corresponds to tumor cells, and the color corresponds to the glioblastoma from which they derive. And in this analysis, the cells are roughly grouped in 4 different clusters, each one predominantly populated by cells coming from a single tumor. Curiously, this is not at all the case when you consider the normal cells that populate the tumor. On the opposite, when we do exactly the same unsupervised analysis, the cells group by their subtypes, their lineage subtype. Like here in yellow, you have all the myeloid cells of the tumor. And in that case, when you look at the origin of the cells that populate this cluster of myeloid cells, you see that they come from the 4 tumors. So this participation of each tumor to each cluster can be objectivized by calculating an index that we call a normalized mutual information index. And when several sources are populating a given cluster as for the normal cells, then we have a very low NMI. On the opposite, when a cluster is populated predominantly by a single tumor, then you do have an elevation in the NMI. This was not unknown, but that was a problem that had never been addressed in the literature. People repeatedly showed that in using lung cancer cells or pediatric glioblastoma cells or melanoma cells, each time they found that upon unsupervised analysis, the cancer cells grouped predominantly according to the tumor from which they were derived, whereas the normal cells were populated by cells, the cluster of normal cells were populated by cells coming from all the tumors studied. So we tried to understand a little bit further what was at the origin of this influence of the tumor on the grouping of the cells. So of course, we first tested differing hypotheses, all related to the fact that you could have some technical bias explaining this observation, although that was unlikely because normal cells do group independently from their tumor of origin. And I'm not going to go into details, but we did everything we could thought of. We could absolutely not remove the influence of the tumor on the cell, of the tumor of origin on the cell grouping by any of these manipulations. So afterwards, we turned towards the tumor-specific biological characteristics like the mutations, their CNVs, and so on. And once again, we tried to address all these possible biological characteristics by different means, and we did not suppress the influence of the tumor on the cancer cell grouping. So that led us to envisage that there is some information that is dispersed throughout the repertoire of transcript of each cells, and that encodes an identity of the tumor from which they derive. And to test that, we just perform what we call a downsampling of the genes that we included in our unsupervised analysis. So we started from all the genes, all the transcripts that were identified at first, and then progressively we selected randomly different numbers of genes and repeated the same analysis several times. And it's only when we started to reach only 500 genes that we began to see a decrease of the influence of the tumor of origin, which was totally suppressed when we analyzed only 50 genes at a time. So that was a problem for us because we could not use unsupervised analysis to extract information about the functioning state of the cells from this unicellular transcriptome. So we turned towards an alternative, which was to reduce the data using molecular signature, this signature being made of genes, and each gene having been experimentally demonstrated to be required to underlie the functional state that we were considering. And here I'm going to show you an example of the results we obtained with a molecular signature corresponding to the tumorigenic state of the cells, that is their ability to initiate a novel tumor. And this signature is composed of 5 transcription factors. So how did we come to these 5 transcription factors signature? By going back to previous work of our lab, but also from other labs, and especially the one of Bernstein at MIT. And some time ago, we were trying to identify the transcription factor networks that are responsible for driving glioblastoma cell aggressiveness. So we designed an assay into which we compare tumorigenic glioblastoma cells and their non-tumorigenic counterparts, and then went on to profile their change in histone mark. And during the course of this process, we identified one transcription factors, RNT2, that was highly actively expressed in tumorigenic cells and completely repressed in non-tumorigenic cells. And looking further into the patients and different data sets, we found notably that RNT2 is rather associated with the proliferative zone of the tumor, and rather than the low proliferative zone. Its expression was also correlated with a molecular signature characterizing cancer cells with stem-like properties. And finally, when we turned toward a biological experiment, we found out that if we knock down RNT2 expression into human glioblastoma cells, these cells totally lost their ability to initiate a tumor when they were engrafted in the brain of immunodeficient mice. When we went a little bit further into our analysis, we found that most of the transcription factors that are downregulated in response to RNT2 knockdown correspond to transcription factors that have previously been shown to participate in the tumorigenicity of glioblastoma cells. we also found for some, we're able to identify for some of them RNT2 binding sites in the regulatory element of their genes. So we selected 4 of them, RNT2, OLIG2, SOX9, PAU3F2, and SAL2, to be components of our tumorigenic signature. As I said, the knockdown of each single of these transcription factor being sufficient to suppress the tumorigenic properties of the glioblastoma cells. So what we did was very simple. We calculated a tumorigenic score, and this score is simply the geometric means of the expression of each of the signature elements. We plotted the distribution of this tumorigenic score across all cells of the dataset, and by using a simple calculation of derivatives of the curve, we were able to distinguish the cancer cells into 2 groups. One potentially endowed with low tumorigenic abilities, and the other one potentially endowed with high tumorigenic properties. And here you just have a PCR illustration of the distribution of cells with low and high tumorigenic potentials into the data set. Of note, in that case, all the different tumors contributed to both groups, either the one with high or the one with low tumorigenic abilities. So we kept going, and we tried to identify the genes that are differentially expressed between these 2 groups of cells. And we essentially identified overexpressed genes. And first, we tried to see whether these this list of overexpressed genes detected in the tumor cells could be observed also in other datasets. So when we started our studies, there was a single scRNA-seq, that one's from Darmanis. So we decided to use the dataset of the TCGA, which gathered the RNA-seq data of 155 glioblastoma tissue fragments. Again, we profile the tissue fragments according to their tumorigenic score, and we took the extreme of the curve, thinking that this group would be enriched in cells with low tumorigenic potentials, and this one in cells with high tumorigenic potentials. We again extracted the genes differentially expressed, and when we look at the overlap between the both datasets, we were very surprised and encouraged by finding this huge 60% overlap between the 2 lists of overexpressed genes. So then a second dataset became publicly available, the one published by the Seva Labs at the MIT, and published in an article whose first author is Neftal in 2019. And this time, this dataset comprised almost 5,000 glioblastoma cells. And again, when we did the same procedure, we were able to find a very important overlap between both datasets. So that encouraged us to extract from these common overexpressed genes the one coding for metabolic enzyme. And we found 66 of them, which were common for both Darmanis and the TCGA tissue, but we had similar results with the Neftal dataset. And when we reconstruct the metabolic pathway to which this metabolic enzyme belong, we found that the cells with high tumorigenic potentials are very much enriched in lipid metabolism pathways. To find out, or at least to try to find out, which of these several enzymes could be key in underlying the tumorigenic properties of the cells, we turned toward the reconstruction of gene expression regulatory network. We reasoned that if this could give us an hint at the most important key elements of the metabolic enzyme by selecting the enzyme located at the most dense nodes of the network. And in that case, we identified an elongase, LOL2, as the enzyme localized at the densest node of the network. LOL2 is involved into the synthesis of very long-chain polyunsaturated fatty acid synthesis. When we look into additional data set in order to comfort the potential interest represented by LOL2 as a metabolic enzyme required for sustaining the tumorigenic property of glioblastoma cells, we had the pleasure to find out that all the variation in LOL2 expression tended to point to its involvement in the control of the aggressiveness of glioblastoma cells. For example, when we look at the lead data set here, the upper graph, this lead data set was developed in 2006, and it is composed of cells extracted from glioblastoma patients that are endowed with tumorigenic property or, on the opposite, deprived of tumorigenic property. And as we hoped and expected, LOL2 was overexpressed in the cells endowed with tumorigenic properties. Next, we tried to find out whether LOL2 was overexpressed in the glioblastoma tissues as compared to normal brain tissue. And here you see the example of the CCGA dataset of glioblastoma tissues and normal brain tissues, which clearly show a huge overexpression of LOL2 into the glioblastoma tissue. And that comes true also when we compare the expression of LOL2 at the single cell level, comparing the cancer cells to the normal cells of the Darmanis dataset. And here, the normal cells mean is here. You have a lot of zero, which are difficult to see on this small graph. So we kept going and decided to test biologically whether LOL2 knockdown could impair or not the tumorigenicity of the glioblastoma cells. So we knocked down LOL2 expression using lentiviral transduction of the cells with an shRNA targeting LOL2, or for the control, a neutral shRNA that we call SH control. So the knockdown worked very nicely in all cells line that we analyzed, and these cell lines are continuously maintained in serum-free medium in presence of growth factors, so in a condition very permissive for proliferation. And despite that, the knockdown of LOL2 expression resulted in a very important decrease in the proliferation of all cell lines tested as compared to cells expressing control shRNA. When we go to in vivo assays, trying to see whether the cells are still or not capable to induce a tumor when they are engrafted into the brain of immunodeficient mice, and we monitor the formation of the tumors thanks to the constitutive expression of the luciferase gene into the cells. So you know that luciferase, if you provide it with its substrate, transforming it to light, so we can follow the tumor development through bioluminescent imaging. So as you can see here, when we use cells with a knockdown of LOL2 expression, we have a very important delay in the tumor induction. And it's even blocked for some other cell line, as exemplified here. The mice never developed tumors. And this translates in a reduced tumor burden, as illustrated here by the measurement of the global bioluminescence value recorded for the control cells and the cells with a down-regulated expression of LOL2. We try to go a little bit further and try to understand how LOL2 could act on a mechanistic level. And looking into the literature, we found that other enzymes of the family of LOL had been shown to be involved in a very important manner in the formation of synaptic vesicles. Knowing on our sides that extracellular vesicles mediate a very important intercellular communication between the cells of the glioblastoma, and that this type of intercellular communication is very active in promoting the growth of the tumor, we tried to find evidence for a role for LOL2 into the formation of extracellular vesicles. So we first went to modelize the different data set we had at the in silico levels. And what we did first is to kind of reconstruct the trajectory followed by the cells throughout the metabolic elements belonging to the lipid metabolism. And as you see here, you have an enrichment. Sorry for, I think I should be able to have a better ear. Sorry for this problem with the pointing. So you can see here that you have an enrichment, a local enrichment of the cells overexpressing LOL2 that, as expected, is superposed and increased in their tumorigenic score, but also the same cells having LOL2 overexpression are superposed with cells that have an enrichment in a score representing all the molecules that are required to generate extracellular vesicles. So when we went on and tried to measure the size of the extracellular vesicles that are produced either by cells expressing a SH control or cells expressing a SH LOL2 and therefore with a decreased expression in LOL2, we find that there was a decreased proportion in the small-sized extracellular vesicles. So that points to a role for LOL2 into the fabrication and genesis and maybe release of extracellular vesicles from glioblastoma cells. And that it is not unexpected for an enzyme that is very important in the fabrication of key elements of the cell membrane. So the first conclusion I want to share with you upon this work is that, as I hope I showed you, there is some information that is dispersed throughout the cell transcript repertoire that encodes the tumor identity and that masks any other information, in particular those related to the functioning state of the cancer cells. That I showed also to you that there is a way into which we can overcome this masking by reducing the data thanks to all the prior experiments that have been conducted by the community and that have already listed a lot of molecules as important for sustaining the cell functional state. We find out that cells endowed with high tumorigenic potentials are distinguished by an enrichment in several metabolic pathways involved in the synthesis and catabolism of lipids. I didn't show it to you, but we were also able to demonstrate that this enrichment in lipid metabolism and also in LOL2 expression can be seen in glioblastoma with varying genomic background. And here I just underline the fact that we can see that in glioblastoma initially labeled as having or not amplification of the EGFR genes. And finally, I hope to have demonstrated to you that LOL2 is a key enzyme required for glioblastoma cell tumorigenicity. All this work has been published at the end of 2019, and it is great responsibility and the great work of a very brilliant PhD student, Mirka Sorti-Serongen. And to insist a little bit more on the biological significance, relevance of LOVL2 for glioblastoma, I would like to say that a very few months before our papers was accepted, there was another paper from Rich Laboratory which was published, and that also highlighted LOVL2, this time as an important molecules for sustaining the stem-like properties of the glioblastoma cells. And what is very interesting is that in this case, they reached this conclusion by starting to identify the super enhancers specific to glioblastoma and the genes that are associated to these super enhancers. And LOVL2 was one of these genes. So as a result, with two entirely different approaches, two independent laboratories converged towards the same conclusion, that LOVL2 is indeed crucial for sustaining the aggressive property of glioblastoma cells, and therefore constitute a very interesting therapeutic targets. So just now I would like to finish to share with you a few progress we made into refining our strategy for identifying the cell functional states of glioblastoma cells in link with their key metabolic underliers. One of the first refinement was to introduce some improvement into our grouping strategy, and that allowed us, by focusing this time on the motile properties of the cells, we were able to identify a very well-defined subgroup of cells which were potentially endowed with either high or low motility. When we compared the genes differentially expressed between both groups, we found out that the genes that are overrepresented in the group of cells with high motility were, by gene ontology, identified like being characterized by a lot of gene ontology terms that all related to cell migration. So the similar data have been obtained this time with 4 independent data sets of glioblastoma single-cell RNA-seq. And all these studies is the object of collaborations with different persons like Thomas Daubon in Bordeaux, who is a specialist of glioblastoma cell migration, Christophe Antoniewski and engineer Léa Bélanger, who are bioinformaticians, and Jean-Michel Perrin in Paris, who in our lab who is a specialist of microfluidic approaches. Just to let you know a little bit of what we came to. The conclusion that so far we have reached. We have been able to implement into our in silico analysis novel computational analysis that allows to model the trajectory taken by your cells from a low migratory state to a high migratory state. And we were also able to extract crucial metabolic enzymes that are apparently located here at the switch between the cells, going from a low to a high motile potential. When we manipulated experimentally the expression of this enzyme, either genetically with shRNA or even pharmacologically with specific inhibitors, we were able to demonstrate with our collections of glioblastoma cells coming from humans that the cell indeed presented in response to the knockdown or activity inhibition of this enzyme a very strong reduction in the ability to migrate upon a substrate such as laminin, or in a microfluidic chip, we even saw that they had almost totally lost their ability to travel through narrow spaces and tortuous spaces that mimic the environment that they encounter when they migrate through the brain parenchyma. So here you see that you have a mixture of cells. The green cells correspond to the cells in which the enzyme of interest was knocked down, and these cells co-express GFP together with the shRNA, whereas the control cells are simply white. So if we look into the microchannels here, you see that most of the cells that are able to enter these channels and migrate through towards the other side are control cells. We encounter only very, very rarely cells expressing the downregulation of the enzyme. And this has been quantified also. And we also verified that the cells with downregulated levels of this enzyme are also impaired in their invasion. Invasion being the ability of the cells to remodulate the extracellular matrix environment. So here we tested collagen, which is very abundant in the glioblastoma extracellular matrix, but also matrigel. And matrigel is very enriched in laminins component that are very close to the composition of the basal membrane of blood vessels. And in glioblastoma, we know that you have several paths of migration for the cells. One of them is within the parenchyma itself, but another one is along the basal membrane of the blood vessels. And as you can see here, you have a very huge decrease of the cell's ability to invade across both extracellular matrix. So as a final conclusion, I would say that our two initial postulates came true. We can identify cell functioning state from single-cell transcriptome, provided we reduce the data using molecular signature. We can also extract the metabolic activities and the key enzymatic players that underlie this functioning state. And that is possible by combining enrichment analysis in metabolic pathway, as well as manual reconstruction, and as well as gene expression network modeling. So with that, I'd like to acknowledge the main actors of our team that participated to this work I presented to you. And first of all, Mirka Sortisi-Rungen, who is going to defend her PhD thesis in the next month. And Elias El-Aber, who was very instrumental for all the metabolism analysis. We also are very grateful to Christophe Antoniewski and Léa Bélanger, who took us towards the world of computational analysis. And I would like to stress out that Mirka made most by herself of the computational analysis that I showed you. And finally, Thomas Daubon, who works in Bordeaux and who is a specialist of glioblastoma migration. Thank you for your attention. And
well, thank you very much. I'm sorry, yeah?
No, I will be happy to take some questions if there are some.
Thank you very much, Marie-Pierre. I'm very excited to see where your work is going to. And they were very exciting data. Thank you very much. So we have some questions here. First, Robson is asking, thanks for a very nice talk, Dr. Junier. Do you think ELOV2, L2 may play a role in another type of cancer?
We haven't had the opportunity to look into that into details, but that's, that can really be the case because it is involved in the formation of lipidic components of the membranes. And we know that from recent studies that more and more cancer types have been shown to be especially addicted to lipid metabolism. So we need to look into more details into the data set that are actually available.
Great. So we have another one here from Marcus. He said, very interesting talk. I wondered what are the fates of the lipids that are synthesized in GBM? does fatty acid oxidation reduce, or they have more lipid droplets?
Actually, fatty acid oxidation is a very important source of energy for the glioblastoma cells. But you still can encounter some lipid droplets, although ourselves, we didn't look at that. But that was reported in more classical cell lines, the ones that are maintained into serum. I don't know, without verifying what is going on with cells maintained in serum-free medium, whether that would still be true or not, because we know that serum brings a lot of things to the cells that are difficult to control, so I don't know. But that could really be the case.
Great. I have some questions too. Actually, I know that you guys work with pediatric tumors, right? So I was wondering if you have like some differences between the pediatric and the adult gliomas, and if you have in these metabolic states that you have showed to us, especially the fatty acid oxidation and the lipids metabolism, and if you have like saw some differences between high-grade tumors and low-grade tumors regarding specifically this metabolism.
So we do not have any information about that. First of all, we believe strongly now that we need to look at the cells at the single cells level, cell by cell, even into culture. So that makes it difficult to look at all the details of metabolism beyond the transcript, because you know that you cannot analyze metabolite projections through fax and so on. Yeah. So there are too many leakage issues. So now what we could do is to look into the most recently available data set on the pediatric tumors, which we haven't done yet, but that could be of interest for sure. But there is, when you look at the profiles of each single cell, just looking at the transcript levels of the elements participating to the lipid metabolism, you see that there is a kind of a continuum. Cells that are very much enriched in these elements of the lipid metabolism, and cells that are very poor in them. So you have this mixture of cells, and in a very active tumor, I would expect all the lipid metabolism, especially fatty acid oxidation, but also very long PUFA synthesis to be very active, because you have a predominance of cells requiring that for their proliferation. But
well, it sounds like glioblastoma because they're very heterogeneous. So you need to. It's expected that you're going to find like these different populations, right?
Yes, right. Yes.
Yeah. And so it's very, very interesting. I am becoming more and more passionate about big data, despite that I'm not very, I don't understand a lot of things, but it's very interesting, and it's being proven to show like some functional states, right? As you guys have shown.
Yes, and even more than that. With our last work, we were able to go from the transcript up to change in a post-translational protein mark modification.
Yeah.
So if you can believe it, it's incredible, the power of this type of analysis. But I would like just to stress out the fact that as a biologist, and I know that a lot of biologists are kind of scared
definitely.
When they are facing this big data and this computational analysis. And I was no different from most of us. But it turned out that it's not that impossible to do, not that difficult to do, especially when, as me, you had the chance to be surrounded by great collaborators and very brilliant students. But in any case, it's very important to try to understand more and more how it works, the limitation of the algorithm, and to have a biologist point of view on them.
Yeah.
Because we found out that with this point of view, we pinpointed things that were not discussed that much by the community at first, although for us it looks very weird. So, and not very logical. So, so that's why it's very important for biologists to not to turn their back towards this big data, but on the opposite, to participate actively to their development and analysis. And besides that, it's me, I am highly grateful to the lab that made this big data set available, publicly available. That's a chance, because they are very difficult and very costly to obtain. And and they come from Stanford, Harvard, MIT. So
we have another question here from Juan Perez Valencia. He's saying, very nice talk. Do you have any clue about the metabolic source for lipid production in GBM? Glucose, glutamine, other amino acids?
That we didn't, I didn't look into all the literature. We do not have any data, precise data for us, experimental data to answer your question. And I'm not very knowledgeable in all this literature. I know it has been burgeoning lately. So, and you have some differences according to the cell lines examined. So I cannot give you, unfortunately, a very precise question, but I may ask Elias, my collaborator, to answer to you. I think he knows more than that than me. Sorry.
Great. Maybe our next speaker will have some inputs about this amino acids. Well, so for now, Marie-Pierre, thank you very much for being here today with us.
My great pleasure.
It's been a pleasure and an honor. And before I invite our next speaker, we're going to make like a quick, brief break, okay? And we will be back soon. So, hello again, everybody. We're gonna continue with our schedule, and it's also a great pleasure to have the next speaker here today as well, Dr. Thales Papagiannakopoulos. I hope I got this right, and thank you very much for being here with us today, Thales. It's a pleasure and an honor. And Dr. Papagiannakopoulos worked at the Dr. Tyler Jack's laboratory at the MIT in his PhD, and he studied the molecular mechanisms that contribute to cancer at an organism level, and using very elegant autochthonous preclinical mouse models of cancer. And he recently pioneered a novel CRISPR-Cas9-based somatic genome engineering lung cancers from genetic modified mouse models. And using CRISPR-Cas9 based genome engineering, he has developed a platform to rapidly characterize the function of genes that are highly mutated in lung cancer and characterize their mechanism of action, and therefore model complex genetic subtypes observed in lung cancer patients. And Dr. Papagiannakopoulos, or Papaji as we like to call him, became an assistant professor at the Department of Pathology at the NYU School of Medicine, the University of New York, in 2015. And currently his laboratory is using genetically engineered mouse models of lung cancer, which can recapitulate the genetics, histological progression, and tissue microenvironment to elucidate the interactions of tumor intrinsic oncogenic events with systemic changes in physiology, diet, and metabolism during tumor progression. And since the establishment of his laboratory, his group made significant progress in understanding some mutations of lung adenocarcinoma, especially concerning the NRF2 KEAP1 mutations. And these studies have led to multiple clinical trials with glutamine-based therapies in lung cancer patients genetically stratified by those mutations, specifically NRF2 and KEAP1 mutations. So, Thales, thank you very much for being here today again. And you're welcome to share your screen with us.
Great. Thank you for the kind introduction, Gustavo. So nice to see you virtually. Unfortunately, I wish I was, I wore my my beach shirt because I wish.
Yeah, great. Yeah, it would be nice to be here in person though. But yeah, we hope that soon we're going to do this.
Yeah. Sorry, I'm just going to look for my presentation here. Where is it? Oh, here we go, Keynote. Can you see it? Does it show up or no? Not yet.
No, not yet.
1 sec. Sorry about that. I have to. Uh-oh.
No problem. Technology doesn't always work as we want to.
Yeah. I just need to do some screen. Okay, I figured it out. It should be fine. Let me see if this will work. Let's try one more time. Share screen. I think it should work this time.
Okay.
There we go.
Yeah.
You can see it now?
Yeah, it's perfect. So put just in presentation mode to, yeah, okay, great.
Got it?
Perfect. Okay, thank you.
Yeah, so thank you for the invitation, Gustavo, and the kind introduction. As you mentioned, my lab is interested in dissecting the mechanism of cancer progression, and we've been sort of going at it from different directions. And although I know a lot of the presentations were regarding metabolism, I will talk a lot about metabolism. But the second part of the presentation, I'll go into sort of a more recent sort of like direction of my group, and that excites me a lot, which has to do a lot with the interactions of the tumor cells with the immune microenvironment. So to put it all together, sort of, we are interested in carcinogenesis, and this is a very complex process, of course, that integrates genetics, environmental factors, physiologic factors, right? So, and really only using mouse models that closely recapitulate the disease progression in humans, we can sort of integrate these different factors that contribute to cancer: genetics, environmental changes, diets, and assess how do they impact tumor progression, how do they impact the tumor microenvironment, immune responses, how do they impact metabolism and systemic physiology. And I'll touch upon all of these today in my presentation and show you kind of how we go by studying these. So we know carcinogenesis starts from early transformation of a single cell, acquires certain mutations. These will lead to a neoplasia, which eventually will, these tumors will become larger, they'll vascularize to get access to oxygen, and eventually you'll get more heterogeneous tumors, regions of tumors that differentiate. So you'll get, as shown here in different colors, tumor heterogeneity. And however, what we do know now that something that occurs during these different steps are various bottlenecks. So tumors also have to overcome certain bottlenecks. They have to evolve to be able to acquire nutrients, to acquire oxygen, to overcome toxic metabolic waste such as reactive oxygen species. And we believe that there's different bottlenecks that occur at different stages of disease progression. In addition to the tumor cells being heterogeneous, also the microenvironment, as I'm sure you've discussed already in this meeting, is quite heterogeneous as well. So you have different types of immune cells coming into the tumors and interacting with the tumor cells. And ultimately, of course, what happens is you'll have metastasis. So some of the aggressive subclones of the tumor will actually metastasize, go to distant tissues and colonize there. So again, these are different stages of disease progression, and there's different metabolic demands that the cancer cells need to have and need to be able to deal with. So our group has studied how mutations that are commonly occurring in cancer, and specifically lung cancer, which I'll discuss today, allow tumor cells to overcome some of these metabolic bottlenecks at different stages of disease progression. But we're also interested in how physiologic factors play a role in disease progression. So for example, our group did some work on understanding how circadian rhythm disruption, so your day and night cycle, impacts tumorigenesis. And finally, we're also very interested in understanding how diets, how dietary intake of certain metabolites, which you have discussed already in this meeting, plays a role in these various stages of disease progression. So our model, and what we primarily study in the lab, is lung cancer. And lung cancer is the major cause of death in both men and women in the US and worldwide. And it's divided grossly in these major histological categories here. And we're primarily interested in adenocarcinoma, which is the major sort of piece of the pie. What's interesting about lung cancer and lung adenocarcinoma specifically is that it's a disease of a lot of mutations, right? So these patients are mostly smokers. And they, because of the smoking being a carcinogen, there's a lot of mutations that occur in these tumors. And you can see this here from the TCGA study. The top in terms of mutation figures melanoma, and right after that is lung adenocarcinoma and lung squamous, right? So this poses a challenge, right? And my background is from genetics, and I always was fascinated with this. How do you actually understand how do these mutations interact with each other, how they impact disease progression, which mutations are functional, which ones are not? So to address this, we were, of course, very happy when CRISPR came out, because that gave us an opportunity to be able to model mutations much more rapidly and much more efficiently. So to do this, to model mutations in lung cancer, we use an existing genetically engineered mouse model of lung adenocarcinoma that takes advantage of a conditional of KRAS G12D and of p53. In these animals, when you introduce CRE in the lungs of the mice by intratracheal administration of viruses, and we use lentiviruses, you can express CRE in the lungs that will activate mutant KRAS by removing this stop cassette here. Now KRAS G12D will be expressed, and you delete p53. Now, this model already existed. The trick that we implemented was we took advantage of the CRISPR-Cas9 system, and in addition of it delivering CRE recombinase in these viruses, we also were able to introduce Cas9 itself, expressed from the same promoter that drives CRE recombinase, and also a guide RNAs of interest. So we can do single guide, 2 guides, 3 guides, right? So what happens now is when you introduce this lentivirus into the lungs of mice, the CRE will activate KRAS, delete p53. The virus, of course, is lentivirus will integrate in the genome. It will now stably express Cas9 and whatever guide RNA you want to express to mutate genes. This was the version 1.0. The version 2.0, which is what we mostly use now, is to take advantage of the conditional Cas9 allele shown here in the Rosa locus. So now, in addition to activating KRAS delete p53, we can also activate Cas9 in the tumor cells with CRE recombinase, and that allows us now to also deliver in smaller viruses multiple guide RNAs of interest. And I'll show you some data where we've used this in the lab. So we've primarily been interested in a specific set of mutations when we started the lab, and this was these 2 mutations here. KRAS, of course, a common oncogene in multiple cancers, a very important oncogene in lung cancer. I told you, and it's the oncogene in the mouse model that we study. But we're interested in this mutation that co-occurs strongly with KRAS called KEAP1. This is a elegant, adapted from an elegant study from Charles Swanton's group, where you're looking at the tumor evolution of lung adenocarcinoma tumors, and they were able to sort of infer from sequencing of tumors which mutations are early, which ones are later, and which ones are really like the late mutations. And the reason this is important is because this allows you to understand whether some mutations are found in all the tumor cells or are found in a subpopulation of tumor cells. The important thing here is that KRAS and KEAP1 are found in the majority of the tumor cells, and therefore may be important for earlier stages of disease progression, and of course may be important because and will be important to target, and when targeting them, we will be targeting the majority of the tumor cells. So as we were studying this set of mutations, a lot of clinical data started emerging from big cancer centers such as Memorial Sloan Kettering, showing that if you have these mutations in KEAP1 along with KRAS, these patients do a lot worse. This is stage 4 patients, where of course they just get standard of care, they do not get surgery. And you can see the KEAP1 mutant patients do a lot worse than the wild type patients. And the reason for this is, one of the major reasons for this is because these patients, turns out, do not respond to checkpoint inhibitors, as you can see here. Checkpoint inhibitors, immunotherapy is the standard of care for lung adenocarcinoma at this point, along with chemotherapy. And in fact, multiple studies have come out recently showing that these patients with KEAP1 mutations are resistant to chemotherapy, radiation, and checkpoint inhibitors. Again, suggesting it's a major clinical problem we need to address. What is KEAP1? So KEAP1 is a negative regulator of NRF2. It binds to it. It mediates its ubiquitination, proteasomal degradation. And what happens is when cells build up reactive oxygen species, or build up certain electrophilic metabolites, highly reactive metabolites, or there's other modifications too. This interaction is disrupted, and now NRF2 does not get degraded anymore. It goes to the nucleus and turns on a battery of antioxidant genes. This is sort of a summary of some of the pathways, the antioxidants that NRF2 regulates. It's really the major transcription factor for regulation of the endogenous metabolic response to promote antioxidant production. So it promotes an ADPase production. It promotes redoxin glutathione metabolism. It tightly regulates iron and heme metabolism. It also regulates multi-drug resistant associated proteins that are involved in detoxification, pumping out certain toxic metabolites or toxic chemicals, including many chemotherapy drugs. So again, NRF2 regulates a lot of different pathways. And the point here is that when you have KEAP1 mutations, what happens is you have KEAP1 loss of function mutations, right? So what happens is now KEAP1 is lost in those tumors, and NRF2 now is constitutively active. So all these pathways are active constitutively when you have mutations in KEAP1. So a few years ago, we showed that when you mutate KEAP1 with using CRISPR, again, somatically, you can actually dramatically accelerate the progression of these tumors. The tumor burden increases And we think based on some of the data that we have that it's early disease progression that's promoted by this inactivation of KEAP1 that now allows an NRF to turn on and suppress reactive oxygen species accumulation through antioxidants. But what's also important is that we showed in a follow-up study that that KEAP1 mutations also promote metastatic dissemination of cancer cells, and they do so specifically through the regulation of NRF, the oxidative related regulation of BAC1, a pro-metastatic transcription factor that actively promotes expression of genes that promote invasion and metastasis. So again, through these KEAP1 mutations, tumor cells are able to not only grow faster, but also become more invasive and metastatic. So now we know this is an aggressive subtype of lung cancer based on our mouse model data. And the human data, we have nice mouse models to study these mutations. So the next question is, how do we target therapeutically these tumors that have these mutations, right? Can we identify novel targets? Like any transcription factor, NRF2 is pleiotropic, and we think that targeting specific nodes, specific genes, specific pathways, one at a time, is potentially not the most effective way to target this cancer, right? You would have redundant pathways here. So our approach has been to really try and identify dependencies that arise from this downstream of this NRF2 antioxidant program being constitutively on. So you have in these cancer cells, antioxidants can produce constantly, right? And that cause some sort of what we call a reductive stress. So these cells are highly reduced. Does that come with a cost? You can think of it as a balance, right? So you have KEAP1 mutations, you have that chronically will promote antioxidant production. A lot of these metabolites that are part of these antioxidant metabolites have high sulfur content because they have a lot of cysteine residues. Does this come with a cost to other metabolic pathways? And how do we identify these? So our approach has been to take advantage again of CRISPR. In this case, the ability of CRISPR to do genetic screens, which are very powerful. And we do so by specifically using CRISPR-Cas9 libraries that target all the metabolic genes that are known to us. We do these screens, and based on pooled genetic screens, now we can identify, are there pathways that are specifically required, metabolic pathways in KEAP1 mutant cells, but not KEAP1 wild type cells? Essentially looking at synthetic lethal interactions with KEAP1. And through many studies now, we've identified that indeed KEAP1 mutations cause certain dependencies. So for example, they cause a dependency in specific anaplerotic pathways. This is the ability, anaplerosis is the ability of cells to feed the TCA cycle, provide carbons for the TCA cycle. These cells are vulnerable to non-essential amino acid, inhibition of non-essential amino acid synthesis and uptake. In work that's ongoing, we have some data suggesting that tumors with KEAP1 mutations are unable to support heme production and therefore are sensitive to heme synthesis inhibition. And finally, in work that's going to come out soon, we showed that they're also exquisitely sensitive to inhibition of specific enzymes in the pentose phosphate pathway through an unexpected mechanism. So all this work suggests to us that indeed, when you have antioxidants being produced, we believe this is a process that's selected for in tumor cells. Antioxidant production is actually beneficial, but it sort of impacts the ability of these tumor cells to effectively and actively supply these metabolic pathways. These pathways work, they're functional. However, they're at a threshold at which if you inhibit them even a little bit, now you can dramatically suppress the growth of these tumor cells. And therefore, these could be new metabolic targets in these cancers, right? So, and importantly, what we're excited about is that this work has now led to multiple phase 2 clinical trials. So these 2 trials are with glutaminase inhibitor. We found that basically glutaminase inhibitors are very efficacious in this KEAP1 mutant subtype because of the fact that again, these tumor cells are unable to feed the TCA cycle. If you block glutamine metabolism, that impacts their growth. And this study here is using a novel glutamine antagonist. And again, all these will be done in the context of tumors with KEAP1 and NRF2 mutations in lung cancer. So we're very excited to see the results of this. This is also important because our work has led to, we believe, to methods to not just target lung cancers with these mutations, but also other cancer types, because these mutations do occur, albeit with lower frequency, in other cancers as well. So, and we have evidence to suggest that targeting these metabolic pathways is efficacious, not just in lung cancer, with KEAP1 mutations, but in other cancers too. And we're also very excited because our work has been reproduced by other groups as well in lung cancer. But as I told you, the groups have shown that in other cancers like breast and pancreas, when you have inactivation of KEAP1 or activation of NRF2 by other mechanisms, these tumors are also extremely sensitive to inhibition of, for example, glutamine metabolism, which is something that our group first identified. So moving forward from this, what we've started realizing is that KEAP1 mutations and KRAS mutations are not alone. If you look at the human genetics, what they tell us is that KEAP1 often is co-mutated with this gene here called STK11, also known as LKB1. And this is fascinating because the co-occurrence is so extensive that approximately 60% of the patients that have the combination of mutations. So if you have KEAP1 mutation, 60% of those patients will also have LKB1 mutations. They're highly co-occurrent in patients. Also, what's interesting is, and this is recent work which has emerged because of the increase of number of patients now that we have that have gone through checkpoint inhibitors, and this in collaboration with Quan Lin's group in Mount Sinai. We now know that the resistance to checkpoint inhibitors is not solely due to KEAP1, as you can see here in red, but is from patients that have a combination of mutations in KEAP1 and STK11, also known as LKB1, as you can see here. It's really the combination of these events that really cause the resistance to checkpoint inhibitors. So our question is why is this, right? Why do these tumors with these mutations, why are they resistant to checkpoint inhibitors? What is it about these mutations that enables the tumor cells to somehow reprogram their immune microenvironment? So to do this, we took advantage, of course, of our genetic approaches and to look at what is the impact of mutations in the tumors and the tumor microenvironment, right, and the immune responses. And to study this, we used the version 2.0 of the CRISPR model that I told you about. So in this case, we have 2 guide RNAs that we can express in the lungs, in the tumors of these mice. And if we express control guides that don't target anything against neomycin, we'll just get tumors that are only KRAS 53 mutant. If we just add 1 guide against LKB1, we just mutate LKB1, 1 guide against KEAP1, or the combination of the two, and then look at how does that impact tumor burden, how does that impact immune responses. So as expected, what we see is that in really early stages of disease, this is only 8 weeks after tumor initiation, we see that the commutation of KEAP1 and LKB1 dramatically accelerates tumor progression. This is the most aggressive tumor genetic subtype we've actually seen in the lab, and that I've actually seen in the literature, period, for lung adenocarcinoma. And that's expected in some ways because we know that KEAP1 and LKB1 are tumor suppressor genes. So you mutate 2 tumor suppressor genes at the same time, it appears that they cooperate. That's great. Sorry about that. Importantly, what we've shown now through extensive flow cytometry profiling using markers shown here, we do usually like at least 12 color FACS panels. We can see now that there's a dramatic reprogramming of the immune microenvironment in these tumors with these mutations as compared to the tumors that do not have these mutations, right? So we're comparing tumor to tumor. And what you see is you have depletion of your macrophages. This is a very abundant macrophage population in the lung. You go from 20% of the immune cells in the lung being alveolar macrophages to less than 5% when you have these mutations. You see a buildup of the interstitial macrophages, a different type of population of macrophages, a depletion of dendritic cells, which are very important for antigen presentation for priming T cells, and an accumulation of granulocytes, particularly neutrophils, right? So, which we'll talk about in a minute. So this is a very extensive reprogram of the immune microenvironment based on facts. How do we dig deeper into understanding what are the phenotypes of these immune cells in more detail? To do this, we do a lot of single cell profiling, as was already mentioned. What we do is we do what's called Cytotoxic, which is a combination of profiling of transcriptome of individual cells by 10x. But in addition to that, we have antibodies similar to FACS antibodies, and instead of having fluorophores attached to them, we have oligos attached to them, barcodes. So then what you do is every cell is coated, is stained with antibodies that bind specific cell surface markers. This cell will be sequenced for its transcriptome, but also will be sequenced for the barcodes that are on the antibodies. And now you can identify cell surface markers in addition to transcriptional expression, transcription in the cells. So I'm just going to talk about the gene expression now. And what we see is, as I mentioned to you before, is when you mutate LKB1 and KEAP1, you go from this amount of neutrophils here in these tumors. You see some neutrophils to a dramatic accumulation of neutrophils, as you can see here. We get several different clusters of neutrophils, which we can see here. So this is quite interesting because people think all neutrophils are transcriptionally inactive. That's actually not true. Turns out you have different population of neutrophils. And interestingly, the ones here on the edge here are these aggressive single GEF neutrophils, high neutrophils that seem to be very pro-tumorigenic based on multiple studies. So now we appreciate that there's all heterogeneity, and we need to dig deeper into this. The other populations are dramatically different, which we knew by fact, but we didn't know the extent of it because you can only really get so much information by cell surface markers and facts, is macrophages. You can see here in control tumors, what you have is this in gray here, light blue gray here, are alveolar macrophages. You can see that these go away with LKB1 KEAP1 mutation. And now what you see is the emergence of these different populations here, which do not exist at all almost in control tumors. These are populations of tumor cells which are high expressing SPP1, also known as osteopontin, which has recently been also identified as a marker of macrophages that seem to be present in aggressive subtypes of multiple different cancers, actually. So based on recent human studies. So we're very excited because now we have a mouse model that actually has these macrophages that haven't been identified in many mouse models so far. So we can really start understanding their function. And we know that there's mutations that promote their accumulation in these tumors. We know these are real because we can visualize them by multicolor immunofluorescence, right? So we have SPP1 shown here in white, arginase 1, which is a common immunosuppressive marker, in orange, and then Ly6G, which stands for neutrophils, F480, which stands for macrophages, and CD11B for myeloid-derived cells, right, which can be neutrophils and macrophages. So of course, what you can see almost immediately is that control tumor is sort of like boring, not much going on in there. You look at the tumors with these mutations, there's a wealth of immune cells in there. It's really fascinating how these immune cells just come in, take over in these tumors and interact with the tumor cells, which is something we're really trying to understand. Now, we know this is actually true in humans because as you can see here, we can stain with MPO, which is a neutrophil marker, or the best that we have for now. And you can see that in tumors with LKB1 mutations in patients now, you can see accumulation of neutrophils, right? So here and here. You can see how the staining looks here in these tumor microarrays. This is a microarray provided to us by Charles Rudin's group at Sloan Kettering. Importantly, these are we have a lot of clinical information about these patients. And what we know is that in these patients that you're looking at here, MPO staining correlates with poor survival. So the more MPO staining you have in red, these patients do worse than patients that have less MPO staining. Again, suggesting that high neutrophil numbers in these tumors correlate with poor survival, which is also what we see in our mouse models, right? So, okay, there's a rewiring of the immune microenvironment. How is this actually happening? How are the tumor cells mediating that? So to do this, we've taken multiple different approaches. We've done gene expression analysis in the tumor cells. We've taken tumor cell condition media from cells in culture. And we're also, importantly, in vivo, collected bronchial alveolar lavage. So this is like collecting, you put through the trachea PBS or saline in the lungs, and then you retrieve it back. And this way you collect whatever milieu of molecules are present or proteins are present in the lungs. And what we have found is that, and this is summarized here, but this is gene expression data, we found that these particular factors here, inflammatory cytokines, are highly expressed in KEAP1 mutant tumors as compared to wild-type tumors. Okay, so we got particularly excited about this because we realized that there was a study showing that one of these, leukemia inhibitory factor, also known as LIF, which is very well studied in the field of of stem cells, primarily, and hematopoiesis, is known to bind to this receptor and activate STAT signaling. Primarily, one of the STATs that can be regulated is STAT3. But this can be phosphorylated, activated, and now STAT3 will turn on expression based on some studies and work ongoing in our group of various pro-inflammatory cytokines such as IL-6, GCSF, CXCL1, IL-33. These have been shown to play various roles in both recruiting and also impacting the activation of immune cells in the microenvironment, or in the context of infection, really. Now, what we found is that you can see here is that when you look at LIF in the bronchial alveolar lavage, it goes from nothing in healthy mice or mice with tumors with no KEAP1 or KEAP1 mutations to this dramatic increase in the lung. And we have also validated that this is true in the tumors by doing in situ hybridization, as shown here for LIF. You can see almost nothing in the wild-type tumors, a lot of LIF in the tumors with the KEAP1 or KEAP1 mutations. And importantly, we do think that this impacts STAT3 signaling, because you can see here, if you actually look for phospho-STAT3 in wild-type tumors, is much lower as compared to what you see in tumors with these mutations, right? So again, we think that LIF turns on in an autocrine fashion, as I'll show you in a minute, stimulates STAT3 signaling, and this now allows these tumor cells to produce all these inflammatory cytokines that impact the immune microenvironment. We know this is actually functional because, again, this is the great thing about CRISPR. We can go now into our mouse models and mutate, delete either LIF or LIF receptor, and look at the impact on tumor growth. First of all, you can see that they both have a very similar impact. Deletion of either has a very similar impact in the ability of these tumors to grow, as shown by MRI here. And you can see nicely in these images how the tumors in these animals with deletions of the LIF or LIF receptor are much, much smaller. They don't really progress much. So the reason what I mentioned earlier, we think is autocrine, is because in this case, we've only knocked out the LIF receptor in the tumors, and that's sufficient to mimic the phenotype of deleting LIF also in the tumor cell specifically, right? So we think the tumor cells are making more LIF. That's causing it to bind to LIF receptor in the tumor cells themselves, and now in an autocrine fashion stimulate this inflammatory signaling. We have a lot more data on this, but I'm just showing you some preliminary data. But we can show here, this is using actually neutralizing antibody against LIF, but we see something similar when we do deletion of LIF genetically. We see the neutrophils go down in these tumors. And also what happens is you start seeing an increase in the alveolar macrophages that had previously gone away with mutations in KEAP1 or KEAP1. So going back to the model, what we think is happening when you use LKB1, you impact the downstream, specifically the downstream signaling cascades that LKB1 is known to regulate. We now have an idea of which ones are involved here. We actually know that the major pathway that's involved in this inflammatory response is LKB1 regulation of SIK1 and SIK3 kinases. We now have genetics showing that if you delete SIK1 and SIK3, you can completely mimic the phenotype that you get with LKB1 mutations. It's the same phenotype in mice. So it's really throughout this pathway, not through other pathways, for example, such as AMPK. And what we think is happening now, this pathway that can regulate CREB specific transcriptional responses can regulate LIF, which then, so you lose LKB1, LIF accumulates, and it will stimulate LIF receptor signaling and the production of all these pro-inflammatory cytokines. So this is the working model. Again, we think that this is actually a very interesting specific phenomenon in these tumors with these mutations. And now we have therapeutic approaches to target this. We use neutralizing antibodies against RIF. The other thing we're trying is we're collaborating with a company that's generated PROTAC STAT3 degraders, so we can degrade STAT3 and see how that impacts the inflammatory cascades in these tumors and their ability to remodel the immune microenvironment. And we're going to try this, of course, in combination with checkpoint inhibitors. Ultimately, we also think that there's metabolic alterations that occur in the tumor microenvironment that can impact also the immune cells, such as T cells. And this is work ongoing in the lab by graduate student Anastasia. So I showed you that we can do genetics and impact the immune microenvironment. How about now seeing interactions of genetics with environmental changes in diets and how that can impact these outputs here, right? So bringing it all together. So, and this with this, I'm going to wrap up my presentation. So a graduate student, an MD student in the lab, Warren, became interested in this question. And to address this question, what he did is he took our KRAS models that I mentioned before with Cas9, conditional Cas9, and he mutated LKB1, okay, and we call these KL, KRAS LKB1 mutant. And what he did is he put these mice in two diets, right? For these mice were approximately 6 weeks after tumor initiation were placed on two diets, either ketogenic diet or high-fat diet or control diet. And what was striking to us was that when you put these mice in ketogenic or high-fat diet, you get exactly the same survival difference, the same decrease in survival. Now, this is surprising to us. First of all, the reason we did this experiment early on was because we thought when you put mice in ketogenic diet, what will happen is, as you'll see here, is glucose levels go down, right? Insulin levels go down. And that's thought to diminish both the glucose uptake potentially by tumors, but also diminish insulin signaling, which can be tumor promoting in the sense that it can stimulate the PI3 kinase AKT pathway through insulin receptor. And you also, of course, a validation that these mice are going into ketosis, you see increased ketone bodies in the blood. So this was surprising to us. We were expecting an extension in survival. Instead, we show decrease in survival. And what's even more surprising is that the high-fat diet had exactly the same effect as ketogenic diet. And the difference, of course, being that in high-fat diet, ketones don't go up, glucose doesn't go down because you have carbohydrates in the diet. So what was also important to note here is that, I'm sorry, this is an error. This should be, I apologize, this should be CDK, N2A, NB. So what Warren did is he took exactly the same model, but instead of mutating LKB1, he mutated INK4AR, also known as CDK, N2A, and B. These are two mutations that also are common in lung cancer. With KRAS mutations, these mutations actually cause a similar, as you can see here, impact in survival in their controlled diet. These are very aggressive tumors. However, in this case, when you put these mice in keto or high-fat diet, there's no impact on their survival. So what does this tell us, right? And these mice do undergo ketosis, as you can see here, lower glucose levels, higher ketones. So what does this tell us, right? This tells us that mutations in the tumor, right, LKB1 mutations, are somehow interacting with the diet the same way with ketogenic and high-fat diet to cause a decrease in survival. So, and what it importantly suggests to us is that it's not just it's the diets specifically, and what is in the diets that's actually similar between these two diets is high dietary fat. So both these diets have a lot of fat in them, of course, despite the fact that they're very different in terms of protein content and also carbohydrate content. We think these mice are dying. So the important thing to note here is that when we take these animals and compare here and compare their tumors, there's no difference in the tumor size or tumor volume. So we don't think that these diets are impacting the tumor growth itself, which is surprising and puzzling, but also very fascinating. So we don't think that the diets are causing the tumors to grow faster, therefore causing the mice to die faster. What we think is happening is that the mice are undergoing some form of cachexia. And one of the reasons we think this is the case is, first of all, as I mentioned to you, there's no difference in tumor growth when put on these diets. And second of all, we only put them, the mice start dying on these diets only after 2 weeks. It's very, very quick. And they seem to drop weight dramatically, even, and they don't gain weight even when they're put on high-fat diet. So you can see here, if you take wild-type mice, shown here, right? And you put them in high-fat diet, they gain weight even after 2 weeks on the diet. That's known. If you take the tumor-bearing mice, the KL mice, and you put them on a high-fat diet, they don't really gain weight, okay, as the control mice do. And when you put them on a ketogenic diet, they also don't gain weight, right? So what we think is happening is that these mice are undergoing some form of cachexia. They're dropping weight quite quickly. I don't have the data here, but we've actually measured weights of various tissues such as liver, metabolic tissues, liver, fat, muscle. They all decrease. And importantly, we see that in the other model that I showed you where there's no impact on survival. This is the model where we mutate CDK into A and B, the INK4AR locus. You can see that they actually drop weight with the ketogenic diet. They become more lean. That can happen in some models. It's expected, but they don't die because of this. And they actually gain weight when you put them on the high-fat diet, right? So again, this suggests that having tumors alone still allows you to gain weight in high-fat diet if you have these mutations. But if you have LKB1 mutations, you cannot gain weight, right? So as shown here. So moving forward, we're trying to understand why this is happening. We think that there's some sort of chemokines or so-called hexokines that these tumor cells with these mutations are making. That's impacting the recruitment of myeloid cells out of the bone marrow, such as neutrophils or macrophages, which is interacting with the diet. So when you put these mice on high-fat diet, now you have these immune cells somehow interacting with the diet, and this inflammation interacting with the diet and impacting the metabolism of these distant tissues. And I want to sort of put this in. We've been thinking about how to sort of like describe this and model this, and I think this kind of reminds us a lot of like what's going on in this in disease tolerance in the context of infection. This is a beautiful review by Metsitov in Science 2012, and this, in their case, this would be viral load here. Okay, in our case, it's tumor burden, where there are certain factors that essentially control whether mice can be sort of, or humans can be healthy, have disease burden, right? They have like tumors or have, say, an infection, and they can tolerate it, right? But there's certain factors that now can, even though you have the same disease burden, can now impact the ability of systemically of the body to be able to tolerate this infection or the tumor burden that's present, right? So the idea is that we think high dietary fat sort of somehow decreases the tolerance, the ability of animals to tolerate having tumors in the lung. So future directions of this work, of course, we want to continue sort of understanding the interaction of how mutations, the tumor microenvironment, systemic physiology are interacting with each other, understanding how metabolic rewiring in tumors plays a role in tumor progression, understanding how immunomodulatory pathways, such as some of the ones I described today, LIF, some other ones that we're studying, IL-33 and SILM2, play a role in remodeling the immune microenvironment, impacting immune responses, and how that may interact with, allow the tumors to interact with distal tissues, right? How the tumors now can interact with fat, with muscle, with liver, right? So we think that we have a nice one with the diet, and that we're going to continue pursuing the high-fat diet. We can also study this in the context of circadian rhythm disruption, something that our lab has done in the past. In both these contexts, and definitely with the dietary fat, we think that the microbiome is playing a role in some of those cachexia phenotypes that I showed you. So that's something we're moving forward we're going to look at, because there's a lot of work showing that both ketogenic diet and high-fat diet can dramatically impact the microbiome in the gut, as you would expect. Going back to the KEAP1 mutations, we still don't know what it is about KEAP1 mutations that's promoting, through NRF2 promoting tumor growth, actually. I told you that this happens, but I didn't know how it happens, right? So one of the ideas is that it may be through the inhibition of the process called ferroptosis. And I'm potentially Rich Bosamato talked to you about this yesterday. But ferroptosis essentially is a cell death process, a cell death mechanism that happens through oxidation of membrane lipids that causes disruption of lipid integrity and cell death. It's an oxidative form of cell death. So moving forward, what we're trying to do is understand that, first of all, does ferroptosis happen in tumors in vivo? I would say this is still unknown. Can we identify markers of ferroptosis, this oxidative form of cell death? And does NRF2 suppress ferroptosis to keep on mutations through activation of NRF2 promote ferroptosis suppression and therefore promote growth in this way? I'm just going to skip this for the time. I already mentioned to you, we're interested in understanding how these pro-inflammatory cytokines impact the immune microenvironment, IL-33, LIF. And again, we think that moving forward, we really want to understand sort of these very complex but interactions that occur. However, we now have nice models to study these interactions. We can sort of like deconstruct them and figure out what's going on, how tumor cells impact through chemokines, cytokines, impact the microenvironment, impact the bone marrow, recruitment of myeloid cells that come to the lung or come systemically into the circulation, how the systemic inflammation now impacts distant tissues and their function, how it interacts with the microbiome to really impact systemic physiology, and this sort of idea of disease tolerance. Again, can we we know high-fat diet seems to sort of decrease the tolerance, but can we identify factors that now will reverse this tolerance? So like and this would potentially be a therapy or some sort of way to prevent cachexia in patients. Approximately 80% of cancer patients experience cachexia, and that's actually a major way by which a lot of cancer patients actually die. So with that, I'd like to thank the people that did the work. Ali and Ray and Anastasia are sort of the resident immunologists in the lab that did a lot of the work that I mentioned to you today in the second part of the talk. A lot of the metabolism work that I mentioned was work done by many people in the group throughout the years. Volkan, Sarah, Warren, Hongyu. Of course, Gustavo was also in the group for approximately a year and a half. So we got to work with him closely, and we're sad when he left to bigger and better things. Thank the funding and of course our collaborators. None of this work would be possible without their help. And thank you for your attention, and I'm happy to take questions.
Awesome. Thank you very much, Thales. A very inspiring talk, as always, right? Thank you very much for your talk. We have a lot of questions here.
Great.
I'm gonna project some for you. So the first one comes from Marcos Oliveira, and he says, "Congrats for the amazing talk. It is possible that high-fat diet and keto diets alters gut permeability and activate immune response that reduce longevity independently of tumor growth?"
That's a very good question. Exactly, that's kind of one of the things we're thinking. And in fact, we're just gonna go down the streets into NYU to our collaborator Dan Littman, who is of course the person that discovered Th17 cells and has been studying the interactions of the microbiome, the epithelial cells in the gut, and importantly the immune cells in the gut, to really understand how that interaction can impact systemic inflammation and impact, for example, immune responses to not just cancer, but also autoimmune diseases, and for example, yeah. So yeah, that's something we're gonna definitely look at, and in fact, we're looking for people to come to the lab and work on these projects. So always recruiting. So if anyone is interested in coming to New York and working on projects like this, get in touch with me. Yeah, but great question.
So there's another one from Hobson. Thanks for the excellent talk, Dr. Papagiannakopoulos. Have you noticed increased systemic neutrophil counts in animals bearing LKB1/KIP1 mutated tumors?
Yes. In fact, we have, and so we do think that they don't just appear in the lung, but they have to come out of the bone marrow where they're generated. So yeah, we do I forget now the name of it. Anyway, systemic blood counts that give you an idea of granulocytes, monocytes, lymphocytes, their numbers, overall numbers, so you can monitor them over time. CBC counts, I think it's called. So we can see as the tumors progress, you see a sort of gradual increase in the neutrophil count in the blood. So they are increased systemically, and that's why we also think that as they are systemically, they probably are also found in other tissues like the liver, the fat, the muscle, where now they can impact the function of those tissues, and on top of that, can interact with the diet to impact to suppress the ability of those tissues to function properly. And specifically, we think they impact the ability of those tissues to switch to fat utilization, right? Which is important in those diets, especially in ketogenic diet, right? You need to be able to utilize fat, and they can't do that. They can't do that in the context of the systemic inflammation. Yeah, and we need to figure out now how to suppress that, right? So that's ongoing work in the lab. So, but we have really exciting data, so I'm really looking forward to what comes from it. Yeah.
Great. We have another one from Gabriela, and she's saying, amazing talk, Thales. Very impressive survival curves comparing KEAP1, LKB1, and currently mutated patients. And she goes on, can you go back to those patients and obtain information on the expression of inflammatory markers? It would be interesting to correlate their inflammatory status with survival.
Yes, exactly. So absolutely, we've done that. I didn't have time to show it to you today. We've done a few things. First of all, we've taken the tumor microarrays that I showed you and looked for neutrophils, but also we've done nanostring GeoMX, which is like a nanostring platform that allows you to do digital spatial profiling. So you can deposit either RNA probes for sort of an in situ hybridization kind of approach, or antibodies labeled with oligos, and assess the abundance of certain cell surface markers. We've used up to 50 now. This now allows us to get information about like co-staining of multiple markers, because a lot of these immune populations are very complex. You can't just look for them with one marker. And we've seen now in patients that you actually you don't just see the neutrophil infiltration. You see that those neutrophils also have high arginase expression, similar to what we see, high PD-L1 expression, similar to what we see in mouse data. But we also see more monocytes and certain macrophages, right? So on top of that, we use these microarrays and stain them for various cytokines, and we see that they're increased. We see that phospho-STAT3 is exactly as we see in the mouse. It's also increased with LKB1 mutation. It's identical to what we see in the mouse. And we've also looked at gene expression data from the TCGA, and we can see there that LIF, IL-6, IL-33, all these factors are higher in patients with these mutations. And in fact, that most of those expression of most of these cytokines correlates with poor survival in patients. So again, we think that these mouse models really are recapitulating what is seen in humans, and we're excited about this because this opens up the door to all sorts of new studies down the line to understand, the interaction with the immune system, their importance in metabolism, etc. So yeah.
Great. And did you see, when you expose those mice to these different diets, did you see if there is any difference between the metastases, if they do more or less metastases?
Yeah, so that's a good question. That's a good question. We don't know. Yet. It doesn't seem so. In the kinetics that we have, it doesn't seem that there's a difference in metastasis. The mice die very quickly because of the diet, so there's no time to. So, but interestingly enough, the model that I showed you, the CDKN2A and B, is a very aggressive model in terms of metastasis. It's actually one of the most metastatic models that we've seen. So now we're excited about they go everywhere very quickly. Yeah. So we're excited because now we have a new model to study metastasis. In that model, the diets didn't have an impact at all on metastasis. So, or survival really. So yeah, but again, it could just be because of the our inability to sort of like study metastasis in the kinetics that we have in these mouse models. But that's an interesting question. Yeah.
Yeah. I was wondering because you showed that there is this secretion of different cytosines, right? Yeah. So it could like have a role in this metastasis. I don't know.
Yeah, because Because, yeah, for example, neutrophils have been shown for many years now, since many years, to promote both angiogenesis and metastasis. They can bring in, they express a lot of matrix metalloproteinases and other factors that can remodel the immune the extracellular matrix. So, yeah, the idea is that when they come in, they promote metastasis. So yeah, it's a good point. And we can actually model metastasis, but we just need to do it in a different way in the mouse model. It's just some tricks we have to do, but that's something we can do down the line. Yeah.
We have another one from Marcus, and he's asking, what are the underlying metabolite signals that converge with reduced longevity?
Yeah, so great question. So it's definitely has to do with it has to be the fat in the diet. Again, we need to now measure what lipid metabolites we have in systemically in these animals. That's something we're doing. But it's unclear. We don't know which of the. So what I can tell you is we've played around with this the types of fat we have in the diet. So the saturation degree. So we've used high polyunsaturated or high monounsaturated or more balanced diets, and we see exactly the same effect. So like it doesn't have its saturation degree of the fat doesn't seem to matter. But yeah, we don't really know. We don't really know what it is about this about the lipids. So this is something we need to figure out. We do have an idea what the factors are that are secreted by the tumors. Obviously, I mentioned LIFNL6, but there's actually other factors that are quite surprising that lung epithelial cells should not be expressing that they're expressing, which is always fun. So we'll see. Yeah.
We have a last one. Is it possible that acetyl groups generated by the keto high-fat diet promote acetylation as a mechanism to increase tolerance to cancer?
Yeah. So it's possible. It's possible. That's something we need to look at. I mean, most certainly there's going to be impact on the acetyl groups in cells, and therefore including in those, not just the tumor cells, but those tissues, right? Liver, fat, all those tissues are impacted, we think, in our phenotypes. So protein acetylation, of course, but also epigenetic regulation of acetyl groups and potentially gene expression, right? So we actually do know that there's dramatic gene expression changes in the liver. We've done RNA-seq, and now we're doing from the livers, and we're now doing RNA-seq from fat and muscle, and we do see that in the livers of animals on these diets, you get dramatic differences in the transcriptome in wild-type mice, but specific ones that happen now in tumor-bearing mice with these diets. So and that is happening probably through multiple mechanisms. Partly it's through the inflammatory responses that come in and perform certain signaling in the liver that can impact transcription, but also potentially the acetyl group availability may be impacting. This is a great question and something we haven't looked at yet, but that's a whole other project. I feel like we're getting something we can do, we can look at in the future. With epigenetic collaborators. Yeah, great questions.
Great, Thales. Thank you very much again. I'm also very happy to see where you're going to, and to see that your models and your studies are generating those new clinical trials, right? So it's very exciting to
yeah, we're very excited. Like, we'll see. Hopefully they work out. I mean, that would be a good proof of principle that what we're doing is can have an impact on patients down the line. So, which is obviously very important to us. So,
yeah. So before you go, I'm gonna invite again our speakers and our organizing committee, so we can have final considerations, right? So, Robson. Okay, so everybody's here. So I'm gonna start thanking you guys, everybody, all of you. It was very interesting yesterday and today. It was very fruitful meeting. We had a lot of discussions, and I open the mic for you.
Okay, I would like to extend acknowledgements. Thank you so much for coming here today, and for the speakers of yesterday also, and hope to meet you soon in other conditions.
Sure thing.
Okay, so I don't want to be redundant, so it was a great pleasure to organize this meeting and receive you all as speakers. We had great talks during these 2 days, yesterday and today. And I should thank also Robson and Gustavo, who conceived the idea. So we thought about joining people working on metabolism together. So it was a great idea, and I think Robson and I just gather together to make it happen. So Gustavo is really a part of this. So thank you all for being here.
Okay, so I guess we.
I just want to thanks. Sorry, just want to thanks for the opportunity. This is my very first time in talking with cancer metabolism, so I really enjoy it, be part of it, and hope to continue interactions with people on metabolism and cancer.
Well, we thank you, Marcus. It was great to pay homage to Dr. Franklin Hojanek, who was a great scientist, and it was a previous oncobiology member it was very, very nice. So, can we finish the symposium? I'd like to thank you all again, and thanks for coming here, and the audience also.
Yeah, sure. Marie-Pierre, do you want to say something?
Just to thank everybody for inviting me and listening to all these great talks. I learned a lot. So thanks again. And the best wishes for success, future success.
Yeah, thank you very much. I hope that next year we're going to be together in person here.
Looking forward to that.
Yeah.
That would be great.
Yeah. Okay, guys. So thank you very much. And bye-bye.
Bye-bye. See you.